Ashish. Uh, Welcome, Ashish. Our first speaker is here with us. Ashish, প্রেজেন্টেশনটা দেখতে চাস? ওই যে একটা নিচের ওই বাটনটা, এই যে আহ, একটা অ্যারো আছে, ওই অ্যারোটা। আশিস, একটুখানি আওয়াজটা একটু চেক কর।
Baladi, when we will start?
Good afternoon. Ma'am, can I start?
I think our honorable speaker is with us. We must proceed, I think.
Shall the YouTube streaming started?
Yes, the video streaming. I'm looking into this YouTube thing also. It is showing live.
Okay, then I am starting. Good afternoon, everyone. I, Bipsha Banerjee, on behalf of Department of Home Science, University of Calcutta, welcome delegates, respected teachers, and all participants to the second International Webinar Series 2021 on the occasion of National Nutrition Month 2021. We have gathered again here to celebrate Poshan Ma. Today is the fourth day of our webinar series. Now I am going to share my screen for a nice presentation. Poshan Ma. Third theme is distribution of regional nutrition kit to Anganwadi beneficiaries of high burden district. The Prime Minister's overarching scheme for holistic nutrition or Poshan Abhijan or National Nutrition Mission is Government of India's flagship program to improve nutritional outcomes for children, pregnant women, and lactating mothers. Launched by the Prime Minister on the occasion of the International Women's Day on 8th March 2018, the Poshan Abhijan directs the attention of the country towards the problem of malnutrition and address it in a mission mode. In order to ensure community mobilization and bolster people's participation, every year the month of September is celebrated as Poshan Ma across the country. Each year, a unique theme is chosen to celebrate the Poshan Ma. This year, as India celebrates the Azadi Ka Amrit Mahotsav to ensure speedy and intensive outreach, the entire month has been subdivided into weekly themes for a focused and assimilated approach towards improving holistic nutrition. The previous themes have already been discussed for the past consecutive days. Today we will be focusing on the third theme, that is distribution of regional nutrition kit to Anganwadi beneficiaries of high burden districts from 16th to 23rd September 2021. Anganwadi means courtyard shelter in Hindi. It is a central area in and around the house where most of the social activities of the household takes place. Anganwadi is a government sponsored child care and mother care center in India. They were started by the Indian government on 1975 as part of the Integrated Child Development Services Program to combat child hunger and malnutrition. We know the popular saying that a healthy mother begets a healthy child. Keeping this in mind, the main beneficiaries of Anganwadi centers are infants, preschool children, adolescent girls, pregnant women, and lactating mothers. Anganwadi workers are India's primary tool against the menace of child malnourishment, infant mortality, and lack of child education, community health problem, and in curbing preventable diseases. They provide services like supplementary feeding, basic healthcare, immunization, education on healthy food, hygiene practices. They also provide preschool formal education to the children, referral services, and healthy learning environment for its beneficiaries. Locally grown nutrient-dense food items can be distributed to the at-risk Anganwadi beneficiaries to combat malnutrition at the earliest. Non-perishable food items that are good sources of calorie, protein, vitamin, minerals, and dietary fiber like rice, puffed rice, flaked rice, soybean, masoor dal, black gram, chickpeas, cowpea, sattu, milk powder, iodized salt, jaggery can be provided to the Anganwadi beneficiaries. The beneficiaries must also be encouraged to grow their own food according to the concept of Poshan Vatikas. Green leafy vegetables, roots, and tubers can be cultivated by the beneficiaries that for consumption. The advantages of distributing raw food grains to the beneficiaries are the beneficiaries can prepare nutritious meal at home. The calorie deficit, protein deficit, and micronutrient deficiency can be rectified. This will help to combat malnourishment and hunger among the beneficiaries. It will help to improve weight gain, growth, immune responses, and prevent anemia as well as other deficiency diseases. Thank you so much. Now I would like to request Purba Chattopadhyay ma'am, our organizing secretary of this webinar, to take over the dais and welcome our delegates. Ma'am, please.
A very good afternoon. Our honorable speaker, Ashish Mukherjee, is with us. We are delighted to have you, sir, with us, and a warm welcome on from the behalf of the Department of Home Science, University of Calcutta. And we are all eager to hear to your deliberations. And Bipsha, will you please introduce sir to our audience?
Yes. Welcome, sir. We are fortunate enough to have with us Dr. Ashish Kumar Mukherjee. Dr. Ashish Kumar Mukherjee is working as Scientist F at National Institute of Cholera and Enteric Diseases, Kolkata since March 2003. His thesis research focused on population and molecular analysis of Vibrio cholerae to gain insights into the interplay between environmental and human disease reservoirs and genetic factors that make certain lineages of this organism highly virulent to humans. His thesis research contributed to around 25 publications in peer-reviewed journals. After completing the PhD, Dr. Mukherjee moved to research with Dr. Douglas E. Berg, professor of molecular microbiology and of genetics, Washington University School of Medicine at St. Louis, USA, and worked for 5 years as research associate. He was particularly interested on the genetic diversity, population genetics, and genome evolution of Helicobacter pylori, and also contributed importantly to our elucidation of mechanisms of susceptibility and resistance to metronidazole, a critical component of combination therapies that are used against Helicobacter pylori. His detailed analysis of virulence-associated genes and housekeeping genes that are under selective pressure showed that Indian Helicobacter pylori gene pool is distinct from the HP gene pools of other parts of the world. His additional studies had contributed importantly to our understanding of the global population dynamics of transposable elements of H. pylori and how they may affect the vigor or virulence of pathogens and HP genome evolution as well. In addition to the contribution to the HP field, He performed detailed, fundamentally important analysis of CTX, prophages, and pathogenicity of islands in a subset of environmental strains of Vibrio quality. Dr. Mukherjee also received training under the Japan International Cooperation Agency project for 6 months in 2006 at the Department of Bacteriology, Nagasaki University, Japan, with Professor T. Hiranama on different aspects of vacuolating cytotoxin of H. pylori. Under his supervision, research students are working as present. Current research interests of Dr. Mukherjee include diversity, evolution, and biology of H. pylori and V. cholerae in the context of molecular pathogenesis, strain evolution, and prevention measures. Recently, he has been very successful in adapting Indian Helicobacter pylori strains to the murine infection model, which will provide further insight into the biological activities of H. pylori virulence proteins and other contribution to the bacterial fitness, and this holds great promise for functional genomic analysis of infection and disease processes. Another important pathogen of recent interest is Campylobacter, a significant cause of gastric illness worldwide, where the research theme is molecular epidemiology, including genomic analysis to provide protected public health. We are very much delighted to have Dr. Ashish Kumar Mukherjee with us. He will be speaking on the topic entitled Reconnaissance of a Enigmatic Gastric Pathogen Helicobacter pylori, Distinct Genetic Traits Therapy, and the Gut Microbiome. Welcome, sir. The platform is yours now. You can share your screen and start presentation. One slide slow mode.
Thank you, Dr. Mukherjee, for accepting our invitation.
I think there is some problem with the audio.
Ashish, there is some problem with the audio.
Screen sharing হচ্ছে কোনো প্রবলেম নেই।
I think sir has some network issues.
I suppose so. Maybe when he joins now, the problem will be sorted out. Maybe.
Can you hear?
Yes, sir. Perfectly.
Yes.
Okay. Window. PPT. So PPT. PPT hয়নি। PPT হয়নি। So দেখুন হচ্ছে না PPT। Can you see my windows?
No, it's not visible.
Your screen, but PPT is not visible. We can see your screen.
Entire screen.
Yes, it is visible now.
Now it is visible?
Yes, sir.
Yes, make it a slideshow.
Yeah, it is in the slideshow.
Then
Enter, sir. Please share your entire screen. Then slideshow mode. Then I will be in slideshow mode.
So it is
not in the slideshow mode. So go to the presentation again, and then then take the entire screen, option entire screen, then you will get the slideshow.
Just a minute. Entire screen. Entire screen. Click share. So now you can see
Yes, sir.
entire.
Entire screen slideshow.
Yes, sir.
Okay.
We can
Yes, sir.
Okay.
Now it's fully visible.
Okay, sorry for the interruption and for nice introduction. So probably I Bipsha, if I don't make mistake. So
Yes, sir.
her name is Bipsha. So okay, so I'll be talking on reconnaissance of enigmatic gastric pathogen Helicobacter pylori, the distinct genetic traits, therapy, and the gut microbiome. So most of the 20th century, human stomach has been considered an environment that is inhospitable for the growth of any bacteria. But the first isolation of a curved microphilic gram-negative bacterium, currently known as Helicobacter pylori, by Marshall and Warren in 1982, had shattered the long-held dogma in gastric microbiology. And they discovered, for their discovery, they received the Nobel Prize in 2005. And after receiving the Nobel Prize during the first news conference, this Australian physician, Barry Marshall, actually recalled about his pivotal shape out of sheer frustration, that how to prove his controversial theory that bacteria are not the trace cause the peptic ulcer. Because before 1982, there was a long-held dogma that the stress is the main cause of gastric ulcer or peptic ulcer or duodenal ulcer. As a whole, we call the peptic ulcer. As I work both on Helicobacter pylori and Vibrio cholerae, just I'm giving a two example that for last 100 years, research on Vibrio cholerae is going on, and around 12,000 paper has come up so far. It was probably 2019 or 20 starters. And from the number of publication for Helicobacter pylori, which has started around 35 years ago, so around 43,000. So from the number of publication, you can imagine the interest on Helicobacter pylori research among the researchers throughout the globe. If I talk about the epidemiology, Helicobacter pylori infects the stomach of over 50% of world population and persists in the gastric mucous layer for decades and possibly for life if it remains untreated. The prevalence of infection varies, but it's falling in most developed countries. Seropositivity increases with age and also depends on socioeconomic status. How H. pylori is acquired and its route of transmission is still unknown or debatable. But since humans are the only known reservoir, it is likely that in developed countries, H. pylori is picked up from siblings, other children, or parents, predominantly via the gastro-oral route. But in developing countries, fecal-oral transmission may also occur. And disease associated with Helicobacter pylori, that it is the only known bacteria which is associated with cancer, and it is the third most common cause of cancer death worldwide. In 1994, working group from WHO had declared that Helicobacter pylori is a type 1 carcinogen. So it is associated with chronic active gastritis, peptic ulcer, gastric cancer, mucosa-associated lymphoid tissue lymphoma. Now, why H. pylori is a chronic infection? One popular hypothesis is that Helicobacter pylori feeds on exudate from the damaged tissue, and much of the damage is caused by the inflammation due to the host response. If it is correct, Helicobacter pylori must work a knife edge. That means it causes enough inflammation for its nourishment, but avoid causing excessive inflammation to protect against clearance directly by inflammation or loss of gastric habitat by damage caused by this inflammation. An infection with one strain of Helicobacter pylori does not protect against superinfection by a different strain. So multiple infection is possible, and multiple type of Helicobacter pylori with different genetic background is also possible. Now the question comes, how Helicobacter pylori survives in the hostile gastric acidic environment, because we know that Helicobacter pylori, our stomach is very acidic. So here I have shown a schematic representation which shows polar flagella, urease, proton-gated urea channel, and production of ammonia. For this purpose, Helicobacter pylori dedicates 7 of its gene cluster for its nourishment, which hydrolyzes this produces urease enzyme, and this is the proton-gated urea channel where urea enters into the bacteria, and it is actually a urease enzyme. It breaks into ammonia and carbon dioxide. So urea, its hydrolysis by urease as a whole produces the ammonia, and that buffers the cytosol and periplasm and creates a neutral layer around the bacterial surface. As a result, it escapes the burnt of the gastric environment. That is, and based on this theory, rapid urease test for Helicobacter pylori has been developed, that where we use the 5% urea, 2% phenol red, and generally if we put the biopsy specimen so into this solution, so it gives this pink color. From there we can understand the person is infected with the Helicobacter pylori. But if it does not show any color change, so he or she is negative with H. pylori. Now, very recently, our collaborative approach has discovered a new player which can shows that adapt chronic inflammation and gastric disease via the pH response BABA mediated adherence. So BABA plays an important role which helps to escape from this highly acidic environment to H. pylori. And H. pylori survives in the hostile acidic environment of the stomach through extensive adaptation using the blood group antigen binding protein A, in short we call the BABA. And this pH sensitive binding mechanism is the key to bacteria staying power and virulence. So that actually researcher from 15 countries have joined together, including our lab, and we have identified a new player, that BABA, very recently, that which is involved actually for escaping the bacteria in the acidic environment. And how they do it? So H. pylori lives in the acidic environment attached to the shell and overlying mucus, where it escapes the toxic effect of the acid in the gastric space. But it faces a dilemma because the cells of the stomach lining have a frequent turnover. As a result, it slid into the lumen, where they cleared up and they slowly goes to the acidic environment. So H. pylori solves this problem by having a pH sensitive addition protein. So this addition protein actually senses that bacteria is going to the acidic environment and quickly detach from the old cell. So bacteria detached from the old cell before they reach to the burnt of the gastric acid environment, and they can migrate back to the new cell where like this from the pH 3. So they quickly go to the from pH 3 to pH 6 area and bind to the new gastric cell line, new gastric cell. And during this time, actually, probably they use the urease for the time being, when urease enzyme to escape from the acid burn, and quickly they go to the pH 6 area. So this was our hypothesis, or in that paper, actually in a nutshell we have shown over there. So now the differences in pattern of Helicobacter pylori associated disease. The duodenal ulcer is more common than the gastric cancer in India, but whereas gastric cancer is more common than duodenal ulcer in Japan and China. So it is known as the Asian enigma. Why it happens, still so far nobody knows. And although 50% of the world population are infected with Helicobacter pylori, it is still a mystery why only 15 to 20% of the infected individuals show the symptoms only. So possible basis for these differences in disease pattern, maybe it is related to the differences in host physiology or genetics, or there is a differences in the bacterial population or genetic differences that affect the geographic pattern of the disease. In different geographic location, different kinds of gastric diseases, or both plays a role. So both the host physiology and the bacterial population genetic difference might have role for the presentation of the different kinds of disease. So for this, actually, we are working on different area on Helicobacter pylori research that I am not going in details. Just few of them I will highlight over here. So first thing, we have worked over here, that are the genotypes of Helicobacter pylori in India distinct. So as I showed you earlier, that how we isolate and identify Helicobacter pylori from the biopsy sample in the blood agar plate. Helicobacter pylori looks like this water droplet, and this is an RUT positive, and Helicobacter pylori gram negative is also oxygenase positive and catalase positive. So if these three things, so the positive things, so then we can actually identify that in the person, the biopsy sample where from where the biopsy sample actually collected, that person is infected with Helicobacter pylori. And also we can do the PCR based assay where we can check for the urease B gene. From there we can identify that if it is urease B gene is positive, so that means that Helicobacter pylori genomic DNA is there. So from there we can identify that person is positive for Helicobacter pylori. Now, the most important part is that Helicobacter pylori is the one of the most diverse bacterial disease. Generally, as I told you, I work on both Vibrio cholerae, E. coli, and other gram-negative pathogens, where Shigella, Salmonella. So there is a definite pattern is there. So when some infection occurs in some locality, we can get so similar kind of DNA finger strains with similar DNA fingerprinting. So here you can see that strains from North India, Eastern India, and Southern India we have taken, and we did not find a single strain which match. So each and every strain from different patient. So you can see that DNA fingerprint for each strain is different. So bacteria is genetically very diverse from one person to another. So that is the one of the major problem for Helicobacter pylori, for fight against problem to fight against Helicobacter pylori. Now we are I'll be showing you that the pathogenicity island of the Helicobacter pylori, that we call the CAG pathogenicity island, the cytotoxic associated gene pathogenicity island. So actually we have, our study has shown that around 90% of the Helicobacter pylori strains contain are positive for Helicobacter CAG gene, but in Europe it is around 50% of the Helicobacter pylori which has that CAG pathogenicity island, but the another 50% they lack this CAG pathogenicity island. That's why they are termed as type 2. So the strains which contains that CAG pathogenicity island, they are termed as a type 1, and the another group is type 2. And in our laboratory, actually, we have checked how we have done it. Just in one slide I'm showing you. So this CAG pathogenicity island actually 40 kb long, around 33 genetic element is there, and 79 pairs of primer we have designed, and there for each genetic locus we have done a certain different PCR, and we have using that PCR assay, actually ultimately it has come out that 82.7% strain has a full CAG PI, and the partial CAG PI, that certain genetic element is present, another and genetic element is absent, in like that. So 9.6%, and the remaining 7.7% are total CAG pathogenicity island negative. And generally we assume that the strains which do not have the CAG pathogenicity island, they are unable to cause the disease. So that why should later on I'll come. So phylogenetic analysis actually shows that Indian phylogenetic analysis of the CAG gene, based on the CAG gene, it shows that Indian Helicobacter pylori intermingling with the European strains, but the East Asian strains like China, Japan, and Myanmar, these so East Asian strains, their CAG gene are totally different from the in India and Europe. And now the gene pool in worldwide CAG pathogenicity island, right end of the CAG pathogenicity island, when we analyzed, just in short, I'm telling that we have identified 3 different genetic categories where we can differentiate the strains into 3 different categories based on their right end of the CAG pathogenicity island. So they are type 1, type 2, and type 3. And this type 3 junction, we have identified a special function unknown zone where which is available only in Indian subcontinent. So that by analyzing certain, doing the PCR, we can tell that these strains had been identified from the Indian subcontinent, from a patient from the Indian subcontinent. Even sitting in USA, when I analyzed a certain biopsy sample without knowing their history or from where the strains came, biopsy sample came, I I identified that these biopsy a group of biopsy sample which I received, they are originated from the patient who are from the Indian subcontinent. So in that way, actually helped us a lot to understand the panmictic nature or geographic partitioning of the bacterial population. Now, another important virulence associated gene, that is the vacuolating cytotoxin. I am not going in details about that. So it has a signal region, middle region, and there is a different allelic form is there. And depending on the different allele, their virulence also differs. So just I am showing you that this is the normal aged 5 to 1 normal gastric cell line. When we treat with the vacuole, it forms the vacuoles within the cell, and that's why this toxin has been named as the vacuolating cytotoxin. And this is the in the cell line. But what happened in the gastric cancer patient, it is the transmission electron microscopy. You can see that Helicobacter pylori causes big vacuoles and the gastric within the gastric mucosa of the gastric cancer, and also it is showing that within the vacuole, actually Helicobacter pylori hiding within this vacuole. And if you try to treat this patient with antibiotics, even majority cases you will be unsuccessful to treat the patient with the antibiotics, because most of the cases antibiotics cannot reach at this niche where they are hiding. And vacuolating site analysis of the vacuolating cytotoxin, also we have seen interesting thing. We have identified that Indian Helicobacter pylori form a distinct character as compared to the ethnic European and the East Asian. So although I have shown that another pathogenicity island, that is the KAGE gene, another virulence associated gene, where European strains are intermingling with the Indian cluster, but here the Indian cluster form a distinct Indian strains forming a distinct cluster as compared to the ethnic European and East Asian. Now again I am coming to the genetic polymorphism at the 3' end of the KAGE. So actually Helicobacter when attaches to the gastric cell line, it's actually releases the toxin KAGE, which actually binds with the using the phosphatase enzyme. It actually binds with the SHP2, and this SHP2 actually is responsible for cell migration and other parts. And this after attachment, actually the Skage H. pylori host cell interaction triggers also later on the morphological change that induced different kinds of growth factors like things that I'll come later on. So this is looks like a beak of the hummingbird. So this actually that's why it is not named as hummingbird phenomena. And translocated actually Kage forms a physical complex with the SHP2 domain with the using the phosphatase activity, and that actually changes the abnormal it actually makes induce the abnormal proliferation and movement of the gastric epithelial cells, and it seems to be the first stage of the first very initial stage of the gastric ulcer, or in future the gastric cancer. And as I'm telling you that it has different kinds of Helicobacter pylori Kage. It is a very Kage gene itself has also a different allelic form, so that I'm coming in details. So if we a similar primer with different type of strains is giving in the 3' end actually Kage gene when we are trying to amplify. So it is giving different kinds of banding pattern. From this banding pattern, you can imagine that they have a different length or structure. And when we analyzed sequence-based analysis we did, we have seen that this just I'm showing you that this red zone, this contains one tyrosine residue, EPIYA. This tyrosine residue is very important because, as I told you, this tyrosine actually residue is required for tyrosine phosphorylation, and that actually causes the cell proliferation, abnormal cell proliferation. So certain Helicobacter pylori we have seen has only one EPIA motif. Certain strain has a 2 EPIA motif. Here you can see certain, so although all are Kage positive, but they have a different kind of structure of the Kage gene, and they contain, so different Helicobacter pylori contains different number of tyrosine containing different number of tyrosine residue, which has the power for abnormal cell proliferation. So person is infected with Helicobacter pylori. What is the structure of the Kage? That is also important. How many numbers of tyrosine residue is there in the 3' end? That is also important. Say for example, this one, they have a 5 EPI motif, and so their ability for abnormal cell proliferation will be much more. And again, this strain also had 3 EPI motif like this strain, but their position of the tyrosine residue within the gene differs, and accordingly their structure will also be different. So several factors actually I am trying to tell you. The strain is positive for Helicobacter pylori, strain is positive for Kage. That is not sufficient. So there are very intrinsic factors resides within the Helicobacter pylori that actually governs the manifestation pattern of the disease. So if we then again just we have tried to analyze the detailed analysis of the Kage 3' end structure, which showed that the distinct and discrete insertion or deletion within the 3' end of the Kage. And if you see that this huge part is deleted in this region over here, and if you insert this part over here, this strain will become like this. So say for example, here you can see that this region is absent, which contains one EPI motif, and again different kinds of insertion deletion has occurred within the KAG 3' end if we analyze the strains. And I'm not describing this one, it will take long time. So it say again the first tyrosine residue, first and second tyrosine residue falls within the beta pleated seed, and the third, fourth, and fifth tyrosine residue again exposed to interact with the host host protein. So as a result, actually again the tyrosine residue so resides within the beta strand. So as a result, it can interact with the host cell. That also differs from strain to strain, that how many numbers are within the are exposed to for interacting for with the gastric host with the host. So and that is also a major important part for precipitation of the disease or for manifestation of the disease. What I was trying to tell, that say for example, if we delete or insert this part, so we can, if we delete this part, this strain will be converted in this strain, and again this this structure we can after deletion we can convert this structure to this structure, and if we insert this part, it will come up like this structure. So in that way the strains can move from one type to another type. So different, and we have our analysis also showed that this area where we are talking about, there is a lot of insertion deletion has occurred. This is actually the hotspot and for recombination, and recombination breakpoint analysis also that established that these areas are the hotspot for recombination, and majority of the recombination event within the gastric niche occurs within this zone. So careful analysis of the amino acid sequence has shown that Helicobacter pylori from India showed some distinct feature present in this repeat region, and H. pylori strain isolated from India carried genes capable of coding proteins which may contain EPI motif as low as 1 to as high as 5 in number, and all EPI motifs are decoded. And also we have identified a 3.4 kb size within the KAG pathogenicity island, and also preliminary data also show our sequencing analysis of this segment did not show any homology with the database. Now I'm coming to the multiple Helicobacter pylori infection in a single host. So for one study we did in 30 duodenal ulcer patient. From each patient, 2 biopsy samples we collected, one from antrum, another from corpus, and from each biopsy sample we have picked up 5 different single colonies, isolated single colonies. That means 5 from antrum biopsy sample, 5 from corpus biopsy sample. That means each patient we have identified 10 single isolated Helicobacter pylori colonies. And just I'm showing one example, and analysis of that single colonies, it showed that 1 to 8 similar kind of pattern when we did the DNA fingerprinting, but 9 and 10 show different pattern over here. And same with another DNA fingerprinting showed that the same with different set of primers. So 9 and 10 showing the different. And when we checked with the KAGE, VACE, and we did the multiplex PCR assay, that 1 to 8, these strains and these colonies actually from that patient 1 to 8, they are positive with the VACE, S1, M1 and KAGE. But here you can see they have the S1, they have the VACE, that is, but they have the VACE S2. That means VACE S2 actually non-functional. That they have a defect in the signal region. So VACE S2, and also they are KAGE negative. So VACE S2 and M2, here the middle region is M2. So they have the VACE region. So 9 and 10, they are devoid of KAG pathogenicity island. So that patient, at least that patient infected with 2 types of strains. One type of strains has KAG pathogenicity island, another type of strains do not have the KAG pathogenicity island. Then when we analyzed the 3' end of these 8 strain, they showed 2 different amplicon. And when we sequenced that amplicon, we found that they have the lower band, they have a deletion of 102 base pair deletion, and within that deleted region, one EPIA motif is there. That means the strain, the person infected with Helicobacter pylori strain, which have Helicobacter with 2 different genetic background within the KAGE gene. So similar thing I have shown over there when we analyze the UDHB or REK of that patient, of that Helicobacter pylori strain isolated from there. So there was a 100% homology with this, but there was a difference in this KAGE region. So there is a micro diversity. Also there is a diversity in the KAGE containing some strains, some colonies have the KAGE gene, some colonies do not have KAGE gene. So in that way, actually, we have found that every patient in Kolkata with the gastric, with the duodenal ulcer, that time when we analyzed with infected, they are infected with more than one type of Helicobacter pylori, and sometimes 5 or 6 times types of Helicobacter pylori. So which one is causing the disease, that is also very important. So always there is a possibility the recombination within this Helicobacter pylori, and they can come up after recombination event. The more virulent strains may come up within this gastric niche, and that time they can cause the disease. So again, I am showing another thing, that when certain Helicobacter pylori we have tried to amplify. This is, as I told you, this is the KAG pathogenicity island. So if we compare, that is the left junction, this is the right junction of the KAG pathogenicity island, 520 to 548. So when we tried to pick up that this pathogenicity, this KAGE gene, using that A and B, if we try to do with the primer, this A and B, this one, we can amplify this one. If we try to this 550 per C and D, say for example KAGE gene, we can amplify this one separately. But when we are using A and D primer for 3 cases we are getting amplicon, we got amplicon, but another 2 cases we did not get any amplicon. So what is the reason? So there may be possibility. So if we get this A and B amplicon, say for example here, so and also C and D, A and D must give amplicon, but we did not get it. So there is a possibility that time it came to our mind that probably in these two strains, their orientation or the two genes are not located in the same location as we are thinking. So just to make a short story, so we did a chromosomal working of the whole pathogenicity island, and we have identified a different structure in the pathogenicity island in certain group of strains. So as I told you, that is this 520 to 548 is the pathogenicity island. So this is the left junction, this is the right junction. When we by chromosomal working of those strains which did not give the amplicon, so when we try to understand the whole pathogenicity island, we found that this part has come up over here, that 519 to 546, that 546 has come up after 510, and this zone 512 to 518, this area has come up over here, and after that one insertion inversion sequence has appeared, and it is around 3.4 kb element. So altered organization of certain genes of the pathogenicity island of certain strains. So we did the colony blot hybridization using, and this insertion, as I told you, that insertion inversion sequence, this is very unique. So using this one as a marker, when we did the colony blot hybridization, we found that this 4 kb element we have used as a probe, and around 20% of the Indian Helicobacter pylori strain had altered organization of the pathogenicity island. But when we did the same analysis with the Japanese strain, these two actually the control strain, but not a single Japanese strain of the 96 we had used did not give any signal over here. So Japanese strain, or especially the East Asian strain, do not have this altered organization of this pathogenicity island. So now I'm coming to the another part, the duodenal ulcer promotion gene of the Helicobacter pylori. So I'm trying to move because many things I have to show. Probably you are waiting for that one also. So plasticity zone, actually another zone of the Helicobacter pylori which is responsible for the disease manifestation, where different this plasticity region can take up many foreign DNA from there, so from the other bacteria. And if you see over here that two different strains, J99 and 26695, these two different strains, only the plasticity region zone over here 913 to 951, this a huge region, actually only 6 genetic element is common between these two. Otherwise, other part is totally different. So only six genetic element, that is this one actually homologous with this one homologous with this one. So that means the plasticity region zone is a very hyper variable region, very much strain specific plasticity region gene of one strain do not match with the plasticity region gene with the other strain. There was a huge difference, and they are actually contain one gene, that is this yellow region, where 917 and 918, if you see over here, there is a mutation over here, and that mutation actually later on actually brings one stop codon over here. As a result, this is actually non-functional. This for this strain, this region is totally non-functional. But if one C is inserted in certain clinical isolates, we have found that one C is inserted over here, then these two ORF actually forms one ORF, and that actually that after insertion it is a continuous, and it forms one ORF. There was no stop codon, and that one actually later on identified as the duodenal ulcer promoting gene. And our analysis in the Indian strain by dot blot hybridization and the PCR, it showed that around 30 to 40% of the Indian strain had a that is a duodenal ulcer promoting gene. And also we have found differences that duodenal ulcer patient with duodenal ulcer, they are significantly contains higher number of this duodenal ulcer promoting gene. And when we mutate the then we mutated this gene DUP from the duodenal ulcer containing DUP containing gene, and relative expression of the islet after co-culturing with the AGS. This is the wild type strain, this is the mutant strains. So you can see that islet relative expression of islet is significantly dropped when DUP is absent. Similarly, islet production actually also in the when it is co-cultured with the AGS cell, also very low over here in that mutant. But if you see over here, and there is no difference in the islet production between the mutant and the wild type strain. Then when we analyzed the DUP, we found that this the DUP of this wild type strains actually non-functional. So when we deleted it, it does not matter. So it did not affect the islet production. Cell cycle analysis, the DUP knockout of H. pylori strains. This is the wild type. It is producing lower amount of apoptotic cell death when we mutate it as compared to the wild type. And also the there was a growth deficient of the mutant Helicobacter DUP mutant Helicobacter pylori as compared to the wild type. And we have later on we have published it. So DUP is and we have found that in the gastric ulcer patient or the duodenal ulcer patient, actually this genetic element 6 times more prevalent as compared to the those who are the plain gastritis patient. Now I am coming to the therapeutic part. So in 2005 to 10, during these times, we have identified that the most Helicobacter pylori strains are resistant to metronidazole but are susceptible to the drug commonly used for eradication therapy and ulcer therapy. Because metronidazole, which is a critical component of the combination therapy against Helicobacter pylori treatment in the other parts of the world, but in Indian subcontinent, this metronidazole, which is the drug of choice for Helicobacter pylori, it is not in the which is a drug of choice in the West, but it is not suitable for the Indian subcontinent, because from our childhood, actually the Indian population takes metrozole or metronidazole for parasitic infection, and that actually which is a lethal dose for the parasite, it is the sublethal dose for the Helicobacter pylori. And there actually one population grows where that sublethal dose of the metronidazole actually brings the resistant metronidazole resistant bacteria are selected in that population, and that as a result around 80% of the Indian Helicobacter pylori strains nowadays are resistant to metrozole or metronidazole. So now why? So that part actually we have worked. So we have worked on different nitroreductase gene. Just I'm telling that one nitroreductase gene, that is the RDXA, actually wild type 26695 strains, it is 1.5 R and 3S. But if we knock down the RDXA gene, then it is becoming 16 or 32S. So that means it is becoming the resistant. Similarly, another nitroreductase gene FRXA, if we inactivate the FRXA, we found that it has no effect on the normal metronidazole susceptibility phenotype. The wild type also showed that 1.5 or 3S, and after FRXA kanamycin cassette FRXA when inactivated, we also found that it is also showing that 1.5 or 3S. So that means FRXA inactivation alone did not affect the metronidazole susceptibility pattern. But if we inactivate both FRXA and RDXA, then what is happening? It is the this strain is 26695 RDXA, inactivated RDXA, it is showing 16R but 32S. 16 resistant but 32 it is sensitive. But when we are inactivating both RDXA and FRXA, so it is showing 32R and then 64S. That means that in FRXA, in inactivation of FRXA, increase the level of metronidazole resistance of RDXA deficient H. pylori strains. So that means both FRXA and RDXA is inactivated in certain H. pylori strains. Actually, it increases the level of resistance power against metrozole. So I'm not going in the summary what I have told so far. Just in a short, very few slides I have shown, and that we have also worked on RDX, another gene. So FDXA, that one I'm not showing for the shortage of time. So now what will be the treatment strategy for as Helicobacter Indian Helicobacter pylori are resistant to metronidazole, that means the metrozole. So combination therapy which is used in the other parts of the world will not be effective in this Indian subcontinent. So generally 2 antibiotics component, clarithromycin and amoxicillin, and 1 proton pump inhibitor we use. But WHO recently had reported that around and also reports are coming from different parts of the globe that this combination therapy, the activity is also 80% or less their effective efficacy rate, because increasing resistivity against this antibiotic, and prevalence of resistance to clarithromycin is increasing significantly day by day. And many studies have proved that the structural change on the 23S rRNA gene is the main reason of this bacterial resistance towards clarithromycin. So this structural change is caused due to the mutation of the 23S rRNA gene. And clarithromycin, which is a macrolide antibiotic that actually binds with the peptidyl transferase region of the 23S RNA of H. pylori and inhibits the protein synthesis. So when there was a mutation in that or single nucleotide polymorphism within the 23S rRNA gene, their structure is altered and it unable to bind in the peptidyl transferase region of the 23S rRNA gene, and it unable to inhibit the protein synthesis. As a result, clarithromycin become that time ineffective. And our study, collaborative study with Amity University had shown that 11.8% strains from North India were resistant to clarithromycin, and our sequencing study also identified A2143 region of the 23S rRNA gene. There was a polymorphism from A to G, that is a point mutation or single nucleotide polymorphism has occurred in the peptidyl region of the 23S rRNA. And then we amplified this region of resistant strains, and we electroporate to the sensitive strain, and later on we transformed colony. We found that they are also resistant with the clarithromycin, and also when we sequenced it, we found that they also contain that the G at the position of 2143 position. So that means that single nucleotide polymorphism which occurred at the 2143 region, so that is responsible for the resistant or emergence of clarithromycin resistant in H. pylori in the Indian subcontinent. And our study serves as a wake-up call to restrict the overuse and misuse of this antibiotic, which is highly effective against many infections. But another problem is that for when we get the patient, when we get the biopsy sample, 5 to 6 days it takes time to get a single colonies, and then we can do that antibiotic susceptibility study. And if we do the antibiotic susceptibility study after 5 to 6 days, it takes another 5 to 6 days to interpret that whether the strain is resistant or sensitive. But this single base point, by exploiting this single base point mutation, we have developed a PCR based method which can detect the point mutation just by PCR. And say for example, we have designed 2 PCR primer, that is forward primer is same, but resistant specific primer and the sensitive specific primer, clarithromycin sensitive specific primer. If it amplified with the resistant specific primer, the strains will, we have checked that they are resistant to clarithromycin, and if they provide band with the PCR with the clarithromycin sensitive specific primer, then the strain will be sensitive with the clarithromycin. And as a result, the day we are getting the single colony, the same day we can give that result, that the strain, whether the strain is the patient is infected with Helicobacter pylori, whether clindamycin based therapy will work for him or her, it will work or not. So it will help to the clinician to actually to guide them, because most of the cases empirical statement actually we give what the doctors among just the common belief, according to the common belief they give the antibiotic prescription, but if we give the result in the same day, they can change within 5-6 days, so they can change the drug regimen now. But still this single base PCR, that PCR method actually takes time, 5 to 6 days, because we have to get the colonies first from the biopsy sample. If we get the culture, so we can the same day when we are getting the culture we can give the result, but because we don't have to wait another 5-6 days. So that's why we have now we are developing at this moment in the lab, the work is going on, that one nested PCR we are developing, where from the biopsy sample, the day we are getting the biopsy sample, we can identify the point mutation of the clindamycin resistance. So in the first PCR, we will amplify the 617 BP amplicon of this of the from the DNA, actually gastric biopsy DNA from the gastric biopsy sample, and then the amplification of the first PCR will be used, which will contain that region actually either sensitive or resistant, whatever may be the response of that single nucleotide polymorphism region, and then this amplified product of the first PCR will be used as a template DNA in the second PCR in 1:500 dilution, and then we'll do the two PCR. One is sensitive allele specific PCR, another is resistant allele specific PCR. So that means the day we will be getting the biopsy sample, the same day after 5-6 hours, we will be able to predict whether the patient is infected with the clindamycin resistant Helicobacter pylori or not. So regions of failure for the eradication of Helicobacter pylori, the incomplete cure achieved with antibiotics because of increasing resistance strains, undesirable side effect, non-compliance, and the cost of the antibiotic regimen, which is not, many people cannot afford it. So, and recently WHO actually H. pylori has been registered by the WHO among 6 antibiotic resistant bacteria that pose the greatest menace to human health. So increasing complication in the conventional triple therapy and stimulate an urgent need to develop a non-antibiotic antibacterial agent which will be effective, safe, and have novel drug targets. So just quickly I'll go through with 2-3 things we have worked. That is, one is curcumin. So major constituent of the turmeric powder, it is well known, and it is responsible for the flavor and the yellow color, and it is used for anti-inflammatory, antioxidant, and anti-cancer. And our study has shown that so different doses actually it can inhibit the growth of the Helicobacter pylori. And also when we infected the mice, we found that curcumin, so normally it is it can infect the mice in colonize, but when we use the curcumin after colonization, if we treat the mice with the Carcumin, we have found that they do not have any growth on H. pylori. And then we PCR product also, we did the PCR of this the genetic element of the biopsy sample of the mice, and we did not find the treated in the treated cases. We did not find the amplicon. From there we can imagine, we can tell that the total eradication of the, because PCR is a very sensitive, and that's why, so it did not identify actually it, as it did not give any amplicon, so it was able to eradicate the total H. pylori from the stomach. And histopathology, if you see over there with the hematoxin eosin stain section of the mice gastric tissue, this is the normal tissue, that is the and when we infect with the H. pylori infected, so there was a damage of the gastric mucosal epithelium. The gastric mucosal epithelium is damaged, gastric pits is damaged, and the gastric gland also damaged. But when we treat with the again with the Carcumin, again restoration of all the parameters occurred over here. So and later on we also found that the Carcumin down regulates the increased level of MMP3, matrix metalloprotease 3 and 9, which actually increased after H. pylori infection. So in I am not going in summary that Carcumin is a can be used as a potential therapeutic agent or for H. pylori associated gastroduodenal diseases. Similarly, another compound we have worked, that is the elegaic acid, which showed promising antibacterial agent against Helicobacter pylori, which is available in high amount in horitoki, blackberries, raspberry, walnut, pomegranate, and strawberries. Here also the similar thing, we found that it is it has a very in vitro, it can inhibit the growth of the Helicobacter pylori, and also transmission electron microscopy, when we grow the Helicobacter pylori, this is the normal control strain, but when we grow the strain with elegaic acid, you can see that coccoid morphology of the bacteria. And also bacterial colonization in the mice stomach, the similar thing as I showed you for Carcumin, the similar effect we have found, that the total eradication has occurred. And the histopathology also showed that these two panels actually shows the negative control, that is and then when we infected with the Helicobacter pylori, we found that significant damage in the mice gastric tissue, but it is again, when we treated with the elegaic acid, it showed that the layer is totally restored. And this like the control, and as compared to the infected, there was much improvement. So both Carcumin and elegaic acid showed immense therapeutic potential by eradicating Helicobacter pylori as well as restoration of H. pylori induced gastric damage. So this study provides novel insights into the therapeutic effect of Carcumin and elegaic acid against Helicobacter pylori infection, suggesting their potential as alternative therapy. And in at this moment, actually, this already we have published this one, and that is one work is going on, that is the gingerol, that which is a compound of ginger, which is responsible for the pungent smell of this ginger. and we have found that it is also very effective against Helicobacter pylori. And also with Dr. Pubalidhar, actually we are planning to work. Actually, initial results are very promising, that is the honey. So I hope and believe we can continue that work, and because our initial study has showed very promising result against honey collected from the Sundarban, which Pubali collected. So, and that is in our pipeline, and that we hope will be working very soon. And that elegaic acid, the work on elegaic acid actually was highlighted in the Hindu newspaper, that a good antioxidant can boost the immunity, and also it can strike, it is able to strike the H. pylori. So now the last part, I have come up just few slides, that Helicobacter pylori and the gut microbiome, that several studies indicated that the gastric and intestinal microbiota may play a critical role in the development of Helicobacter pylori associated disease. However, whether Helicobacter pylori can function as a bacterial driver or and interact with the other gastric bacteria, which are subsequently involved in the process of different gastric diseases or finally carcinogenesis, that remains unknown. That is totally unknown. Very recently, 2 studies have been published that highlighted specific microbe in the gastric and intestinal microbiota linking to H. pylori infection and related gastric diseases. One study came up from the Rajiv Gandhi Center for Biotechnology, Kerala, India, under the leadership of Shantanu Chattopadhyay, which is a very good friend of mine. So what they have done it actually, they have taken endoscopy of 375 persons, and in the Trivandrum Medical College and Rajiv Gandhi Center for Biotechnology, and they analyzed the vaccine and the vaccine, as I told you earlier. So genotype of Helicobacter pylori strain isolated from different individuals, they have done it, but they did not find any association between the because as I told you, that just mere doing that kind of genetic analysis, present, absent, these kind of things will not give you association against the severe disease versus mild disease or the asymptomatic individuals. But when they analyzed the microbiome by Illumina MySec, that showed the relative abundance of the abundance of the Bifidobacterium in the lower gut is significantly low for the H. pylori infected patient with peptic ulcer or gastric cancer. Here you see this green zone is the percentage of the Bifidobacterium. The person infected with gastric cancer or gastric ulcer, there is a huge low percentage of Bifidobacterium, but with the person with non-ulcer dyspepsia or just simple gastritis or H. pylori negative or gut positive H. pylori negative, so they have a Bifidobacterium in a huge proportion as compared to significantly higher proportion as compared to the person infected with H. pylori and have the gastric ulcer or gastric cancer. So there was a significant difference in the proportion of the Bifidobacterium between the disease patient versus asymptomatic. So the question comes, having a bad bug. Yes, obviously Helicobacter pylori is a bad bug also, so it is responsible, and also lacking the good bug. So whether Helicobacter, so it is also important to understand how Helicobacter pylori actually kind of dislodge the Bifidobacterium. So the disease is causing due to the eradication of the good bacteria or dislodging of the good bacteria, the Bifidobacterium, or the proteins which they are releasing that actually causing the host cell damage, or the toxin they are releasing actually damaging the bacterial population, that is bacterial population existing in that person. So that is also another important part. So the result of this study provides clues for developing early prognosis of severe gastric diseases and also to formulate effective probiotic that confer protection against these diseases. That if we can identify certain effective probiotic which actually helps to restore the Bifidobacterium percentage within the individual, so we assume that the person person will feel better against the gastric diseases. Because the next study what I'll show, that another study from China which showed elucidated that the effect of Helicobacter pylori on gastrointestinal microbiota. So what they did, major findings of this study, that so what they have done, I am not going in details, that microbial dysbiosis index revealed that the dysbiotic microbacteria in Helicobacter pylori positive mucosa was associated with advanced gastric lesion and intestinal metaplasia. But if we eradicate the Helicobacter pylori, this dysbiosis index is getting changed. So the change in fecal microbiota also included increased Bifidobacterium after successful H. pylori eradication. So if they eradicate, so they also did the treated the patient with the antibiotics and eradicated the H. pylori, and slowly the Bifidobacterium percentage has been restored. So increased Bifidobacterium percentage has been occurred. So that two very important papers has come up, which actually shows that H. pylori infection contributes significantly to gastric microbial dysbiosis that may be involved in the gastric carcinogenesis. And successful H. pylori eradication potentially restores gastric microbiota to a similar status as found in uninfected individual, and shows the beneficial effect on the gut microbiota. And findings from this study provide new insights into the microbial mechanism of H. pylori associated gastric carcinogenesis and the new evidence for beneficial effect of the prevention strategy as a whole. So, should we eliminate Helicobacter pylori from everybody? Because 50% of world population are infected with Helicobacter pylori. So, should we treat everyone? Martin Blazer, one very eminent scientist in USA, so he said that this bug might protect against the chronic backlash of stomach acid into the esophagus called the GERD, gastroesophageal reflux disease, and its complication, including a cancer called esophageal adenocarcinoma. Angry critics charge this unproved theory, if put into practice, could harm the patient. Because after knowing all these things, if we tell this kind of things, so it will harm the patient. And another person, David Graham from Baylor Medical College, he actually told that calling Helicobacter pylori beneficial is like carrying a hand grenade in your pocket with the pin pulled out and saying that it protects you. So just I showed you, and so this is a very debatable issue. Should we eliminate Helicobacter pylori from everybody or not? But one from my research, I can tell that if someone is infected with Helicobacter pylori and suffers with different gastrointestinal problem, he must be treated with antibiotics for eradication of Helicobacter pylori, because here actually 70 to 80%, around 70 to 80% Indian population are infected with H. pylori, Helicobacter pylori. And when and how will develop the disease, or when and how will develop the symptoms, that part we don't know at this moment, because disease prognosis is very slow and depends on many factors, and when and how will show the symptoms maybe 10 years, 20 years, 30 years, 40 years, that we don't know. Even at all will be showing the symptom, that also we don't know, because different factors. Some factors I have tried to collate in my talk over here, but besides that also, if you want, actually if you discuss with me, I can show many other factors, so that actually governs the disease pattern, disease manifestation. So it is a very complex disease, and relationship with Helicobacter pylori with the gastrointestinal problem as a whole, as I can say at this moment. And last 5 years actually we have published on Helicobacter pylori around 20 papers, if you are interested. So and also I have collaboration with different scientists on our lab has a collaboration nationally and internationally, and I am specifically marked red the Dr. Pubali Dhar, that I am really interested to work on the Sundarban honey, because as our preliminary results are very promising. So hope in near future we'll be starting the work. And these are the H. pylori that group in my laboratory, who actually the past and present members. So what I have presented, the work, all of them actually are contributed in different way. And also at the end, I want to acknowledge ICMR, DBT, CSIR, and also NIIT Japan, Okama University Japan, Emory University Atlanta USA, and Johns Hopkins University for providing different kinds of fund for different kinds of research actually, not only Helicobacter pylori, that is also I work on different other areas also. So at the end, thank you so much for your patient sharing. I have taken a long time. So I'll be happy to take some questions from you.
Thank you so much, Dr. Mukherjee, for such an informative session. There are a few questions for you. With your permission, can I start, sir?
Yeah, so just I want to come up. Okay.
Sir, recent studies have indicated that the gene for cell viability of pylori is the target for its treatment. Can you highlight the chances of this gene presence in the Indian population?
Can you repeat the question again?
Yes. Sir, recent studies have indicated that the gene for cell viability of pylori is the target for its treatment. Can you highlight the chances of this gene presence in the Indian population?
Okay, the genes responsible for viability, that actually, again, there is a lot of diversity, even the in different population. So viability of Helicobacter pylori also differs from strain to strain even. So that is obviously there is a lot of work is going on for the for targeting that genes to develop some antibacterial agent. So but so far actually in Indian circumstances, we nobody, I'm not sure that how many people has have been able to do anything, but other parts of the world actually obviously people has done a lot and they are working on that. But in India, very few people have worked on it, and that actually, that part actually very still so far I can, in Indian perspective, I can tell you that is a gray area and it is a really important area for further research.
Okay, sir. Sir,
Yeah.
next question. Is it possible to utilize symbiotic combination of Bifidobacteria and prebiotics to develop therapy against Helicobacter pylori?
Yes, yes, yes. So actually, as I told you already, I have told you that prebiotic and bifidobacteria that can be used as a uh, prophylactic. But the problem is that you have, if someone is infected with, as I told you, that infected with Helicobacter pylori, if you and show the symptoms different, and if you find that bifidobacterium concentration has in the gut microbiome has reduced, so in that case supplementation of the bifidobacterium will obviously give some good result. But at the same time you have to target the Helicobacter pylori also, because bifidobacterium will not able to kill the H. pylori. So you have to eradicate. For treatment also you require that will kill the Helicobacter pylori. So eradication therapy and the supplementation of the bifidobacterium, it will help a lot.
Okay, sir. Sir, in Indian subcontinent, which antibiotic resistance pylori infection is seen widely?
In Indian,
In Indian subcontinent,
yeah.
which antibiotic resistance pylori infection is seen widely?
So as I told you earlier, that is the metronidazole combination therapy that is widely common. The metrozil, which is actually as I already discussed very in great detail, that Helicobacter for metronidazole, why we metrozil will not be able to use against Helicobacter pylori in the Indian subcontinent, because for parasitic infection we use a lot from our childhood, and many people take just 1, 2, 4 for any kind of antibiotic infection. So and the when we are taking that antibiotic, so that metrozil, the metrozil is also very mutagenic. So certain, as I told you, the RDX and FRXA gene, which is nitroreductase gene, it so it causes mutation actually in that RDX and FRXA gene of Helicobacter pylori. As a result, the dose which is lethal to the parasitic infection like amoeba, that is the sublethal dose for Helicobacter pylori. And within this sublethal dose, it causes mutation in the nitroreductase gene of the Helicobacter pylori, and that that mutation in the nitroreductase gene actually helps to increase the resistance power against metrogene, and as a result, in the Indian subcontinent faces a great challenge against metrogel. Actually, the metrogel we cannot use for Helicobacter pylori treatment, which is very effective in other parts of the world. But still, we can use the clarithromycin and amoxicillin combination. But again, that 10 to 15% patients showing nowadays showing that the Helicobacter pylori are resistant against that clarithromycin, as I already mentioned to you.
Yes, sir. And next is what is the mechanism of interaction between Helicobacter pylori infection and host dietary factors? How they can be used for therapeutic intervention?
Yeah, so lots of actually dietary, as as I told you, the two element already I have discussed, and those are say for example, very recently we are working the Carcumin I have told you, ellagic acid I have told you. So those compounds which are available in a great amount in so say for example ellagic acid, the horitoki, blackberries, pomegranate. These are the we can use as a bigger amount for our regular consumption if we take. So it will obviously it will help, and also not only that as I told you, the ginger, honey, that again. So lot of uh, mm, uh, our day-to-day life in especially in India and other parts of the world. So we can use many things for the treatment or betterment or of our health as a dietary supplement.
Okay, sir. Uh, Sir, can cancer-associated abnormalities be reversed by eradication of Helicobacter pylori in host?
Yes, that that is once actually depending on cancer. Actually, it has a it it will take a long time actually if I want to discuss. So it depends on the stages in which stage you are. So certain stages when it is certain population or certain patient reach to a certain stage when H. pylori eradication will not be effective for the treatment for gastric cancer if already developed gastric cancer. So if it is in the beginning stage when so then that time it is, and it is on not only the Helicobacter pylori, it is a complex network actually. As I told you, so different as you have seen in the total gut microbiome, the homeostasis or the balance of the gut microbiome is getting destabilized when a person actually developing the disease, and especially the gastric cancer and gastric ulcer or duodenal ulcer patient, they are developing they are in their case the homeostasis of the bacterium, overall gut microbiome is getting destabilized. As a result, so when we are treating, that is also important, in what stage actually. So that part, so depending on that actually, whether if it is developed a gastric cancer, so so again that depends on the stages. So if it is a very initial stage, that Helicobacter eradication of Helicobacter pylori and treatment with different probiotic or different bacterial supplementation of different good bacteria, that may help. But once it progress further, so only eradication will not at all be useful that time.
Sir, which is the latest combinational therapy employed for Helicobacter pylori infection?
As I told you, that is the clarithromycin, amoxicillin, and one proton pump inhibitor.
How does
Hello, sir. I have a question in this therapy. It has been a long time we are using this double antigen and one anti-secretory
Yes,
drug, but recently I have seen they are using bismuth along with this one. Can you highlight on this one?
yes. The because as in those cases, actually, clarithromycin resistance, the patients when they are coming back after 3-4 months, they are coming back with the same problem. So that time when we are checking, so they have developed a group of bacteria. So initial stage, they do not have probably that 23S mutation or something, that as I told you, but the incomplete doses of antibiotics actually sometimes generates the antibiotic resistant bacteria within the host. And that time, so when they are coming back, so the same antibiotic is not possible to give, that is the clarithromycin amoxicillin combination, because that already that time clarithromycin is resistant to that bacteria. And in that case, actually bismuth will be very useful. And as a result, bismuth is giving, yes, alternatively that time bismuth is giving, and that is giving a good result, at least for eradication of Helicobacter pylori.
Okay, thank you, sir.
How nutrition can help combat Helicobacter pylori infection?
How?
Nutrition.
That means how the diet,
Diet.
how we should
Therapeutic
make the diet when a patient is suffering from Helicobacter pylori.
Okay, so diet part, as I don't know, so that is you will be very so diet as a whole, that is that diet supplementary, actually you can tell, that is what we have already discussed. So for example, that is that say for example ginger, and also I have seen even certain other compound that actually
Are these patients admitted to hospital?
which patient?
H. pylori, patients suffering from H. pylori, are they so? Is it a very fatal disease or like that?
No, that is when it will be a gastric cancer patient, you can
Okay.
you can imagine. So that's
Oh.
otherwise they do not need to get admitted, but it is a nagging problem. So
Yes.
throughout the year, once somebody is started to suffer with the gastrointestinal problem, so whole day he or she will be feeling very bad kind of things. So
Okay.
the patient who actually, though those patients, those have been infected and suffers from that kind of problem, they only know that really that how actually irritating the problem is that, because they will not nausea, vomiting kind of thing. So in the always feeling that the heartburn kind of things. So
Is there any study with post probiotics, that is compounds from probiotics that are released from the probiotics? Is there any study on the
so
effect of whether these compounds can fight against Helicobacter pylori?
no,
Is there any such study?
always there is a actually doctors or studies whatever it is going on, that is with antibiotics some probiotic. So unless you eradicate the H. pylori, as I told you, for time being you may feel better. If
Okay.
you certain period of time you give the probiotic and other things, but the H. pylori has a power to destabilize the homeostasis again.
Okay.
So
It
you
will again grow.
it will again grow because it has a different niche. So they will not go away from that niche unless and until if you give some antibiotic therapy. But at the same time, so already they have made some damage in the gastric microbiome. So already they have destabilized. So if you give some probiotic treatment, so the patient very quickly again come back to the normal gastric microbiota, and they will feel much better or normal life, and they will come back to the normal life.
Okay, sir. Sir, is this Helicobacter pylori infection hereditary?
No, not at all hereditary, but problem is that if say for example if a father or mother is infected with Helicobacter pylori, generally we have a bad habit actually to kiss the baby. So when and Helicobacter, if someone is infected with Helicobacter pylori, so even their saliva sometimes also contains the Helicobacter pylori. So from there it can go to the children, and also sharing the foods. So one if you somebody is eating something and that food actually they are the mother um, or children taking the same food together, and sometimes the same using the same hand after taking food, we do not use the spoon in the Indian subcontinent. The same even if we use the spoon, the same spoon we are using, what I am using, I am giving to my children the same spoon with another with another pool of food, and then the saliva from is transferred from myself to the children. So in that way actually transmission from the parents to the children occurs. So or within the family members occurs. So it is not hereditary, but if we don't become cautious with our health hygiene, so it can transfer within the family member.
Sir, my question is,
Even from the maid servant, even some studies are there. The maid, maid servants are the responsible to transfer the Helicobacter pylori from the from them to the other family members.
sir, what preventive measures should we take to decrease the spread?
What preventive measures?
Should we need to take? to decrease the spread of this infection?
Yeah, that actually no. So as I told you, several we have to maintain that good health hygiene. So sharing of foods and using the same glass and other things. So, and major problem for Helicobacter pylori infection is that it is a chronic infection. It is not the acute infection. So when and how we are getting infected, so we don't know even. Say for example, today after going after giving this lecture, when I'm going out from the lab, I'm taking some food from the street. So if I'm infected with Vibrio cholerae or Shigella or Salmonella, the within 5 to 10 hours I'll show the symptoms. But if I infected with Helicobacter pylori, nobody will know. Even I'll not show the symptoms tomorrow or something. So when and how you have been infected, you don't know. So only thing that you have to maintain good health and hygiene, and also the stool sample. Say for example, here the the sewage is the major source of different kinds of diseases. So stool sample also contains a lot of Helicobacter pylori. So in this part of this world, that is the fecal-oral route actually is the one of the major source. Even when we are taking, say for example, that say pani puri in the street corner. So many they are using, and from actually it has been shown that from them also many bacterial diseases can transfer from that person to the others. So and that because those are the acute infection, I'm talking about the acute infection, but they can be a potential source for chronic infection also. Because acute infection, you will be knowing that if I take pani puri today and if I felt if I develop some symptoms in the night or tomorrow morning, I can imagine that from there, that is the source. But if I infected with Helicobacter pylori, that part I will be not knowing. So, but they will be also the potential source. So that health and hygiene is the major part.
Okay, sir. Sir, one last question. Can stress induce the Helicobacter pylori infection occurrence?
Can stress?
Yes.
Stress actually no, can do many things. So it will not increase the Helicobacter pylori occurrence, but it can because H. pylori infected persons, the it can aggravate the situation as a whole. I can tell that because many persons are infected with Helicobacter pylori, but only a certain population.
Yes, sir. That is my question. Actually, about 75% of the world population is having this organism, but only few get the infection. That is why I'm asking, if we are in stress condition, can it get induced that infection prevalence?
No, yeah, that's I'm telling. So the whole my whole talk was lying on that area actually, that so many factors are involved for this precipitation of the disease or manifestation of the disease. So even I did not highlight the even the host factors. Certain host factors also also have a role for the disease manifestation, for this disease manifestation. But if stress can release lot of things actually. Say for example, acid secretion, that is also increases during the stress condition. And not when there is a lot of acid secretion occurs during the stress condition, H. pylori actually, and that actually causes some damage in the gastric duodenum, and that in that duodenum area generally H. pylori, when H. pylori binds in that, once they get the niche of the damaged area, so this damaged area they can again uh, increase more damage in that part. Generally they are staying under the mucous lining, but when the mucous lining, when ulcer is formed, that means in that area mucous lining is not there. And once they get the chance in that, without mucous lining in that area, they can colonize in a better way. So again the more damage can occur over there. So stress can always a additive factor, obviously.
Thank you, sir, once again. It is our immense pleasure to get you with us. Thank you, sir.
Thanks a lot to
Thank
all
you,
of you for
Ashish.
patient sharing.
You can stop your presentation.
Okay, thank you.
Thank you, sir. It was very nice hearing you. After so many years, I am hearing Helicobacter pylori from someone. Thank you so much.
Okay, it's my pleasure.
Now I move on to our next session. Dr. Amlan Chakraborty has already joined with us. Welcome, sir.
Yes.
Yeah, very good evening to all of you.
Yes, sir. I would like to introduce Dr. Amlan Chakraborty. He is a full professor of information technology in the A.K. Chowdhury School of Information Technology at the University of Calcutta. He is also the Dean, Faculty of Engineering and Technology of his university. He is an MTech from the Department of Radio Physics and Electronics, University of Calcutta, and did his postdoctoral research at the Indian Statistical Institute, Kolkata. He was a postdoctoral fellow at the School of Engineering, Princeton University, USA during 2011-2012. He is the recipient of DST BOYSCAST Fellowship Award in Engineering Science in 2011, Indian National Science Academy Visiting Faculty Fellowship in 2014, JSPS Invitation Research Award in 2016, Erasmus Mundus Leaders Awards from EU in 2017, and Hamid Visiting Fellowship from Cambridge University in 2018. He has been associated with reputed international and national institute of repute as a visiting professor, to name a few: University of Cambridge City, University of London, University of Oradea, Romania, Sunny Buffalo, USA, GSI Helmholtz Research Laboratory, Germany, University of Bremen, Germany, CERN Geneva, Kyushu Institute of Technology, Japan, etc. He has received multiple project grants in the area of embedded system design, VLSI design, quantum computing, cybersecurity, and computer vision from various national and international agencies. He has published around 130 research papers in referred journals and conferences. He is a senior member of IEEE and ACM, Secretary of IEEE, CEDA India chapter, and Vice President of Society for Data Science. He has more than 215 research publications. His areas of research interest are quantum computing, embedded system design, VLSI design, computer vision, and analytics. It is a great opportunity for all of us to hear Dr. Chakraborty on the topic entitled Technology Assisted Nutrition Analysis Towards Food Security. Welcome, sir. The platform is yours now.
Yeah, thank you, Bipsha, and thanks to the Dean of the Faculty, Professor Kubali, uh, the, and all the distinguished
Thank you. that
Yes.
you have amalgamated so distinct, so different two topics in one platform. It's really nice. I think we will hear a great talk.
It's a challenge for me too. That means I am not a nutritionist, or I am not also an biology expert. I think my previous speaker has a lot of expertise in this domain. But what I have recently started working, and that I will try to uh, try to tell you in my lecture, that this is a recent activity what I have started, which is technology assisted food security and food safety. And I will give a little glimpse of that activity, what we are doing with the University of Florida, and also a couple of industry partners in our university. It just started. Right. Before going into that, that means, as I know that I also am learning this particular domain of what is nutrition, a little bit of this, right? I may sound a little bit very naive in terms of the nutrition concept, what I will say. uh, so don't look at that, but try to my the takeaway from my today's lecture for the students who are aspiring nutritionists, nutritional science researchers, right, and also the faculties, will basically the interdisciplinary activities what I am trying to focus upon, and that I will wish to engage all of you in that particular journey of my research. Okay, so without wasting much time, I will start my presentation, and I hope that everything will be fine, because in this particular scenario we are we all have to accommodate these technological glitches as well as promises. Okay, so I hope my slide is visible to all of you, right?
Yes, sir.
Yes,
It's visible.
Okay, so this is the title of my talk, right? I am a hardcore technology researcher. Okay, so I will try to look into the areas where technology can be pushed through, right? And because we also have to continue our research, and I've recently found that there are a lot of things are happening in the nutrition analysis, and towards food security. But before getting into that, I will try to give you a little bit holistic view of what is this technology assisted nutrition analysis, right? So uh, so this is a scenario. I think you all are experts in this domain. So planning for nutrition, normally what we see is a very standard type of suggestion which comes from just if you check the internet, or if you try to check some data, that what are the good nutrition requirement, and how the nutrition requirement can be supplemented by food. These are the very standard information you get, right? Uh, there are also some type of marketing which is coming, which is in a great way, right? People are pushing the nutrition products, because people are very sensitive to nutrition, right? Because everybody needs food, and everybody wants to have good food, which keeps him or her healthy. So a lot of propaganda and a lot of investment is there, right? That means if you go to the Western countries, I think some of you know that the push is more rigorous. Okay, different types of dietetic plan, different types of gluten, non-gluten, ketogenic, all the types of terminologies have been used, and I am a zero in all these terminologies. Okay, yeah, I'm not an expert, but this is one of the push which is coming up, which motivates us. That means, hey, this is a particular domain where something some knowledge already exists, and can we use technology somewhere, in this process. Now the challenge what has come up is that, and where we require data, right? Where we require data, and where we require try to capture the data and analyze the data and try to develop some sort of analytic engine is the personalized nutrition. Because from my layman knowledge of about human health and nutrition, right, I know that every individual is different, right? Every individual has a different so liking and disliking of the food. They are in different health condition. They're in different nutrition requirement level depending on their age, depending on the activity what they're doing. So the process, which was basically a gross nutrition analysis, right? Can we go towards the personalized nutrition, right? And that is what is very important. Now in a country like India, it is more important, right? We know that that means we can easily say that a person having who is a rich person, right, who takes very costly food, right, each of his or her meals, whether he is good in the in terms of nutrition content, maybe not in some cases. Similarly, a person, right, who is just taking the basic food material, not very fancy foods from the different stores, but he or she is just preparing the food, and very basic food, right? Whether we are whether satisfied with the nutritional content, it may or may not be again, right? Because maybe he or she is missing out some of the nutrition content because due to the non-availability of something, right? Due to the cost of the food, right? So what I mean to say here is that this variability cannot be addressed just classifying the population, right? Hey, you are a rich guy, right? You are mediocre and you are poor, so automatically I will see that you have nutrition satisfaction of nutrition requirement is good, bad, or worse. It's not like that. Actually, it's not like that simple maths, right? And I know that you experts, you know all these things. So how to bridge this information, and the most important why I focus on India, right? Because India has a large variety of population, large diversity of population, right? And that's why with the variety of population, the variability in the nutrition requirement, right, and nutrition supply, right, is also quite a varied, right? So this is one of the picture what I want to put to all of you, right? If we try to focus on individual nutrition, then the thing is, what do you mean by that, right? How we identify an individual, right, and how we map the individual to the nutritional axis, the nutritional feature space. Maybe the feature space may not be a very well-known term for some of you. Feature space is something by which we analyze the data, right? Suppose I try to analyze the a particular type of fruit, right? I try to extract its its color, its shape, its smell, right? And I tell that what type of fruit it is. Similarly, a person also needs to be classified, right? What type of nutritional requirement he or she requires, right? For that, we have to first of all understand what are the features to reveal this nutritional requirement, and these are the type of the things what I get from the standard research, right? It tells that it's a demographic data, it's a lifestyle, anthropometry, dietary practices, and eating habits. So this all needs to be collected in some way, right? And it's very difficult, right? It's okay, fine. It's not so easy, right? And what I will try to say is that means how you do that, how you do this process, right? So that you reach the first goal. The first goal is that you need to understand the nutritional features of an individual, right? And considering the larger sector population in India, it's a challenging task. Okay. So there are certain techniques by which we can that mistake the phenotype. Phenotype are the external expressions, right, of your body. It's called the phenotype, right? It's maybe the parameters of your blood. That means your metabolic activity, your microbiome, where we can actually analyze the different components of the body in the micro level, right? And there are there are many processes, right? You can do a microfluidic study, which is a very high technological type of test, which is now very common, right? You take that means you take a sweat drop of sweat, or you take a drop of blood, right? And you can actually react that with lot of components or reagents, right? And you can find the intensity of those reactions, right? And from that you can that means you can try to find out that what are the constituent components, right, of those body fluids, and from that you can try to get to know that about the health condition, about the nutrition condition of the particular person. So microfluidic is one of the particular way to do that. So these are some of the things which are coming up in the technology side, and you can explore. All of you can explore this, and this is coming in different forms, right? Previously, it was a very a packaged type of stuff, but now the microfluidics are coming on paper, on different types of polymers. So people are using that. So one way is that, obviously. Second way is that obviously the genetic data, because genetic data also has an influence on nutrition, as I have found from the literature, right? Again, just to tell you, it's not that that means I am the expert of the domain, but from the literature I saw that there are works which are going on where they are trying to analyze the gene and trying to find the demographic source of the particular individual, and try to understand what type of food habits that individual has, and try to understand the nutritional requirement. There are biological processes by which we can do that. Now how we can incorporate, or how we can bring the new technologies here, right? The support technology, support mechanism to access this data, or to try to capture this data, right? There are a lot of smart applications, right, where we can give the user an interface, right, to put the data, right, or to connect the test data with the device itself. A microfluidic can be connected with your smart device, right, and microfluidic results can get into the device, right? Your many of the test data can be accessed to the cloud, right, and the device can connect to the cloud and access all your test reports, right? And then it can also take some of your instantaneous data from your with a very, very, very useful say app, and that data can be collected, right? Though it may seem very fancy, for a person who is not so well acquainted with the technology. So this may work in the people who are in the city, or who are well acquainted with technology, but what will be the way you can do this for the larger population of a country like India? That's still a big question, right? This not a well answered question still now. There are certain study we can do, right, which can be quantitative survey, right, and which can through quantitative survey, which we can actually generate the qualitative data, right? So some of the very interesting survey which are reported, right, in this particular journal, right? It's a data which tells that what are the expectancies of personalized nutrition. So it was done with male and female in a particular age group, not in the all age group, right? And they have a little bit different type of philosophy, right? So so the mail was okay. So the survey from the mail was more on the, hey, if somebody okay, somebody takes my data, okay, my dietary data, right? What is the consequence? That means whether data will be used by the other government agencies to track my monetary power, to track my locations or something like that, right? So right, and another the other data was actually what are the okay, that means the data can contribute okay, to understand my different types of metabolic activity or calorific activity. So there are different perceptions on the nutrition data which was done and shown in this particular thing. So what I mean to say here is that if you tell me that I want your nutrition data and you don't tell me what's the actual reason you are doing it, I will try to perceive in some way, right? And my friend can perceive in some other way. Okay, gender type A can perceive in some one way and gender type B can perceive in some other way, right? So this is also a sort of because nutrition data or my health data, right, mapped with the nutrition are not very common data for many of the individuals, right? So understanding the context is a bit difficult. Second, there are some social aspects here, right? So when we try to think about nutrition or when we try to think about the food, there are different opinions. So one opinion is that means, okay, it was again a survey done between and these are all data reported by the public data reported by this journal. So when we look into the data of that means, suppose I tell you to actually put your data okay, for taking the lunch or something like that. Okay, so now a particular population or a particular population of will try to know that what are the food I want to take in the different intervals. What are the easy way of food availability, right? The other motivation may be that whether this food can serve for all the members of the family. Okay, so suppose you offer a nutrition okay, suppose a food for a particular population, okay, different groups of the population can have a different reaction to this particular food, right? And this is due to the social context. The third is that means how you offer the food, okay, to the persons, right? Somebody offers a personal touch, okay, that means you go to a place and you try to try to go to a place and you try to take a food where you can be comfortable and when we can take a comfortable food, right? That's all for you. But some else try to see that here what all others are eating. Okay, they are more biased towards the food which is mostly taken by their friends or something. So this is also the very interesting thing that means when you are trying to capture the food data and okay, the eating habits of a person and trying to assess the nutrition, right? You try to push the supply of that food to the particular individual. Different groups of populations have different types of perception on all the aspects from the food choice to the periodic okay, the period of taking the food, and again taking the food from a particular delivery interface. Okay, so these are the variability in the data set and the philosophy. So it's very difficult to actually cater on uniform or one single box for everyone, right? And that's the challenge in terms of nutrition analysis. What I try to say here is that why I say I'm not very expert about every bit of this particular features of nutrition, but what I have understood is that the way you offer the food, the way the data for the food and going up to the nutrition is not a uniform perception for everyone, right? And that's the challenge, right? How collect the data then, right? Or how you use the data to serve, okay, to serve the community. Eating environments is also different. Where you like to eat, right? So that's also a variability, okay? So now how to do this, right? How to sort of personalize nutrition is a big challenge because it is a large variability, and that was very well proved by a lot of research activities. So the suggestion or the push which came is that you need to record the data, because there is a high value. Whenever there is a high variability, you cannot just do a random sample and try to tell that, hey, this is the process, right? You have to take the sample across the various sections of the population, right? And so personalized monitoring and personalized data capture is one of the issues, and the way it can be done is that extrinsic. A person can fill the data manually to the interface, or there can be some automatic capture processes, like you can capture the data from the microfluidics data. You can capture the data from your smart watch, which maintains your metabolic activity, calorific activity, your body activity and all these things, whether you're working or whether you are sleeping, whether you're taking rest. All this activity profiling can be done, and that data can be captured. So this is one of the good way of taking the technology in this particular scope of activity. Okay. Now data analysis, right? There are different types of psychological traits on this eating index, right? And there are different types of eating index. Okay, some are like the healthy eating index, Mediterranean dieting index, and this actually has a different type of scores across the different populations and different point of time, right? And also there are certain factors, like the health factors needs to be considered, right? Your some of your habits needs to be considered, right? And this to be considered as an independent variables. So there are different types of ways by which you try to analyze the data, and then you try to classify the data. Okay, right? And that is the those are very 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 important aspects of the data analysis. There are a lot of data analysis tools, right? If you look into that, right, you can have a sort of classification. Okay, you can have a sort of dietary input in the data, okay, which defines your the eating plate. You can give the physical activity, you can give the medication, right? You can give the different types of information from your condition of your different organs, right? You can trap your activities from the wearable device, and all this can club and can be defined as a big data problem, right? Why it is a big data problem? A big data problem, I think those who have a little bit understanding of data analytics, they know what is a big data. Big data means when the data comes from multiple sources. Data coming from different modes of the data are coming. That means they're describing different windows of the same problem, right? So okay, it's a large amount of data because we are dealing with data of a population, right? Of a human population. So whatever human population we try to consider, whether it is in the regional level, state level, country level, it will be a large amount of data, and we are trying to take the data in a some periodical fashion, right? The data of a person in a different seasons across the year can change. So it's so the volume and the variability of the data is really makes it a very big data problem. Okay, and from this analytics we can now have a precision nutrition. Okay, precision nutrition means we can now actually target to the nutritional requirement right, which can look into all this data, and can have a extended feature space, right, and can target this, so that we can have a precision nutrition, which means that we are trying to fit the individual with the proper nutrition, okay, not more, not less, okay. So that's the direction, but this is not a very easy problem, right. It can that means I can just put this in one particular slide of my PPT, but I know that this is a big problem. I think all of you who are expert in nutritional science, they will appreciate that, that understanding this and making a model out of this, and trying to either do clustering or classification, and trying to generate the precision nutrition is a challenging task, okay. So health inequalities is also there, and this has been also shown that that there are some some good contributions, uh, for this benefit has been visualized or seen to the personalized nutrition, right. uh, so the process, what is being understood is that your health information, your nutrition information, and your socio-economic environment, there is an intersection, right. Your health or your genes are the intersection with your nutrition, right. What you need to have depends on your health, right, or it may be majorly guided by your genetic pattern, right, or your genetic background, right. And also there is an environment, right, in which environment, whether you're in a working environment, or whether you are in a rural environment, whether you're in a city environment. So there are overlaps, right, on this particular thing, and you have to consider all this to fix up your nutrition, okay. So I'm not getting into this. There are different types of ways. There are different types of applications have been come up, and people are targeting the younger people because they are more the working group of the society, right, and they need to be nurtured good way. Okay, it's not that people who are like semi-aged or a little bit older people from my age, they don't require this type of support. They require this type of support, but the way you can provide the technology support to the younger generation, right, it's a little bit upscaling task to provide this type of technology support to the people who are in the higher age group, okay. So there are different types of applications are there, right. You can give your personal information, you can give your health information, right. You can try out these apps which I have shown here, and you can get a sort of personal nutrition, right. I cannot validate the outcome of this because I am not an expert. I will just suggest you, okay, that means if any one of the student can help me, okay, that means to go through these apps, or a group of students can help me to go through these apps, right, and try to find out that how much these apps are really helpful, right. It will be very good for me for my project, okay. So I'm just skipping all these things. So this is the pathway, right. So when we try to do this, when you try to generate a technology space, okay, in this particular nutritional analysis, right, there are certain steps, like conceptualization, development, testing, and then it can go to the market where any user can interact, right. And this is a still challenge. So now on this challenge, what are the technologies available, right? The technologies available from we start from the agriculture as our source, right? Though it might not be directly connected, okay, with the nutrition, okay, directly, but in our project scope, okay, we are taking the data directly from the agricultural stuff, okay, because that is the source of our food, okay. So So IoT, which is the Internet of Things, I don't know whether you have some idea of that. These are tiny sensors, right? Which can be either, okay, they can collect the data of your agricultural source. They can collect the data of your nutritional requirement, like your smartwatch, right? Your smartwatch has several sensors, right, and it directly communicates with the cloud environment, or your smart devices, which means that, okay, the term Internet of Things means that normally when we tell internet, it means computers. So computers can connect with the network and can exchange the data through the internet. But at the present generation of technology, or the present era of technology, it's not computers who can only connect to internet and exchange the data, right? There are devices, there are sensors which some nodes, okay, can actually capture the data through the sensor and can push the data, right? So that is a really a good technology to capture the data. Okay. Now, now there is something called smart infrastructure, which I'm again not getting into because I feel that the core concepts of information technology, computer science, and not the thrust area, okay, in this particular seminar in such a way. But I will just try to tell you that these are some of the terms, right, if you're interested, because why I am saying is that, right, if you take the artificial intelligence, though artificial intelligence is something which is mostly covered in the computer science course, right, it is not that artificial intelligence means only computer science. Artificial intelligence can be an umbrella of any science, technology, or social science. There are many physicists, many biologists, okay, many medical experts, right, who are doing fantastic artificial intelligence. Okay, maybe in some cases better than many of the computer scientists, because artificial intelligence is such a thing that it works on logic. So these are the technology handles, okay, we are trying to explore, right? So artificial intelligence means that whether I can have machines which can act like humans. This is in a very simple way I'm telling, but it doesn't mean, okay, it doesn't mean that the artificial intelligence depends, okay, in a very deep way is only that, but it's actually in a very simple way the machines which can work as if they have the human intelligence, which means that they can understand, they can identify, okay, they can react, okay, to the change in the environment, right? And if we can do the machines in that way, then so suppose, okay, give me a scenario. So suppose you have an elderly person at your home, right? And he or she requires some medicines. Okay, it's not all the time. He or she requires medicines in some special period of time when there are some body parameters which goes higher than the normal, right? And you don't have any physical person, okay, to actually diagnose the problem, consult with a doctor, right, and then take the prescription of the doctor, go to the store and try to give the medicine to elderly person. That means either elderly person is living alone, right, or elderly person cannot move in that way. So there are fantastic solutions which are already existing, though they are pretty costly now. You cannot actually give it in a plenty to all the people, but there are solutions available where this elderly person can have a sort of bed, and the bed has some sort of wearable sensors, right, and the sensors collect the information in a periodic interval, and there this sensor data is communicated directly to the doctor, right, and the doctor gets on his or her smart device gets an alarm if some of the body parameters of one of his or her patient is not in a proper condition. Then the doctor can immediately switch to the scenario by video. There are video cameras, talk with the particular person, and then the doctor can trigger a process by which a particular pharmacy store will deliver the medicine and send a nurse to the particular patient, right? So in this process, actually the human intervention is very less, but all the devices are communicating with each other, right, and taking some decisions that, hey, this is the point of time the device is to send the data. This is the point of time the data goes to the hospital or to the doctor, and doctor initiates a process by which the data goes to the pharmacy, and the pharmacy initiates a process by which the drug is transferred to the person. So this type of activities are now technologically viable, right? It is not done in a very large scale because still the cost is not in that way, but within 3-4 years of time, we feel that these are the things which are coming. So these are the contribution of artificial intelligence, right? Apart from searching the drug and trying to analyze the COVID data and trying to find which drug will work good for the COVID virus, those are also activities which are being done by artificial intelligence, because you cannot explore all the intelligence unless you actually go into the path of artificial intelligence. Cloud computing gives you the fabulous way of storing the data, right, and offering the service, right? We are very well connected with the services like Amazon, like Big Basket, like Groffer, like so many to name. So how they are offering the services, okay, there are so many parties are involved in each of the activities, right? It is not that all the parties have a single web application. It's not like that. So it's a web application between multiple parties which are integrated to the clouds, right? So whenever we have multiple service providers, right, you cannot make them in one window unless you have cloud. Okay, so cloud is an interface which has multiple connectivities, right? And through the connectivities, okay, X can access to Y and Z services, whereas simultaneously B can access to M and N services, right? And all goes seamlessly, right? And there are big giant cloud service providers. Okay, edge computing is a little bit very upcoming. Okay, many of us still don't know what is edge. Okay, but this is also something like a miniature cloud, okay, which are done at the doorstep. So what it means that means you have some Wi-Fi router, right? You have some network switches, okay, you have some LAN switches at your office, at your laboratory, at your home, right? Those can also work as computing nodes, right, when you require a very fast service, right? And that's the concept of edge computing. Okay, so this is the technological scenario which we try to maximum exploit, right, and try to see that what good we can do, right, in terms of nutrition analytics and going for food supplement. Okay, so now I just hit the bullseye about the activity what we are doing, right? So the previous one was actually the just to set up the context. Okay, so our main goal here is that food safety and security. So food safety means that whatever food you are offering, okay, providing, right, it should be good quality food, right? It should be having good nutrients, a good quantity of nutrients. It shouldn't be adulterated. It should not be stale. This is called food safety. Okay, and food security means that you are trying to work in such a way such that every individual of the population gets his or her needed quantity of food. So though they are a little bit different in their motivation, food safety and food security, but this makes a very holistic path or a very holistic type of service to the society. Okay, so we're using all the different type of services. How we can collect the data which are being from the food preparation preparation sources. Okay, there are a lot of food are prepared in the restaurants, in the hotels, in the households, right? Somebody can use a service, like a smart device that's okay. Say, hey, I have just made 4 plates extra food, right? And I don't want to get it in the West, right? Because I don't want that. I will take the okay food next day, because that might not be my motivation, right? My motivation is that, hey, whether this particular extra food can be served to the people who requires the food, right? So the process is that means you need to have a sort of aggregation service. It's called aggregation service, when you have the multiple data from multiple sources comes and tries to churn out some some decisive data. So you need to have an aggregate sort of aggregation service, and aggregation service aggregate the data of the excess food, be it hotel, be it household. There is a service by which anybody can report that, okay, this is the time I prepared the food, and now it is 4 hours from that, and now I have 4 plates of this food left, and can you help me to distribute it? Now this service will actually go to the data, get in the data, and will also try to see the requirement. The requirement will be generated from lot of social welfare groups, right, who are working with this particular type of population, right, who are underfed, right, who are actually who are unable to have the proper nutrition. So can we connect the two, right? And that's the figure which is being shown here. We're connecting these two to a technology platform, right? It's a challenge. We know that it's a challenge, but it's not doable, right? If we have the proper technology platform, if we have the proper mindset, okay, we can do it. Okay, that's one of the challenge we were going to solve in this project. Okay, it's obviously storage is also one of the factor. That means a lot of the food is wasted because it's not stored well, right? Can we actually try to find that, okay, this is a food type A, this is a food type B, this is a food type C. Okay, food type A should go to storage type 1, food type B should go to storage type 2, right? And can we decide this quickly, and can we have a very robust management, so that the foods are properly stored, right? So they can be preserved and they can be used for future use. Okay. There are some, that means, as we know that there are good people and there are always people who do wrong things in our society, right? And okay, that means, and okay, we are unluckily, that means there are a lot of cases which has happened that people have deliberately have actually provided the food which is not good, okay, for their own benefit. How we can prevent this? But how we can prevent the quality and the quantity of the food can be assured, right? So a person or a particular food source is trying to push the food through a particular chain to the population which needs it. Now there are some persons in middle, right, who has a very wrong motivation, right, of actually jeopardizing this particular activity. So there are data security techniques, okay, which are called blockchain, and some processes can be done, okay, so that we are also looking into that, how technology can be used for tracking the food adulteration in the fast way, modifying the food storage data and food supply data. Okay, and those modifications cannot happen if we have a sort of data authentication procedure, okay, and okay, through the blockchain. Okay, so that is these are all new technology, and we're trying to use the technology, okay, for the solutions. I'm not telling any solution at this point of time. Okay, I'm just trying to provide you the landscape, okay, or the motivations, right? So the focus is that, okay, and the last but not the least is that is to analyze the nutrition information, right, to wearable devices, to periodical monitoring of the population, right? And this is again a very challenging task, because the technology support cannot be provided to a large number of population we have, right? And how to generate low cost technology, okay, how to generate the technology which is affordable, so that the nutrition analysis can properly done, right, is one of the core issue of this particular problem, because otherwise we cannot have the basic nutrition requirement data. Okay, I'm just skipping. This is another part of this. Okay, decarbonization, which also has an effect on nutrition, our health. This is another side of it, but I'm just skipping this. Okay, not a core part. Livestock management is one of the also issue, because majority of our food, some of our food like milk, okay, okay, like the meat comes from, it comes a lot of food stocks, okay. Okay, a lot of poultry products, okay, are there, and we have cow for the milk, right, and for the all the milk products. So we are trying to also monitor the health environment of this particular livestock, which is also an important aspect of the total ecosystem, right, and also the greenhouse gas effect due to the methane release of this particular, okay, from this particular animal. This is a very focused project of a lot of this environmental, okay, from environmental end. Okay, long-term solutions are something that whether we can have a sustainability, because whatever solutions we try to do, right, whether we can have a sustainable solution, either for the production of the food, supplying of the food, using the services of the, or the data of the food gets aggregated and distributed, right? How to engage the NGOs, and how to actually, ultimately food has a very good connection with the psychology, right? I feel very happy, all of us feel very happy if we have a very good lunch or dinner, right? And that makes us to generate a very happy sense of the family. So that also is something which we need to keep in mind. Okay, so now how all this will be done? Okay, this may be a big question, and I invite the experts from the nutrition department, and I just throw this in this platform that we are in the process of generating a center of excellence on Internet of Things. This has been thematically approved by our Honorable Vice Chancellor. Okay, and we are talking with multiple parties so that we can set up this, and food safety and food security will be one of the core aspects of the activity of Center of Excellence. This will probably a multi-campus facility. Maybe we can have one arm at the nutritional sciences group at our Baliganj campus, and maybe the technology arm can be at our technology
we
campus.
have already the work is going on the civil work is going on for this.
Excellent. So this is there, and the team is something like that. We are having an academic part. The University of Calcutta will be in a strong way. University of Florida has already given their consent to be a part of this activity. Jalpaiguri Government Engineering College, which is a government engineering college of okay, in the northern part of our state, which is a very strategic region in terms of food, in terms of cultivation, right, in terms of crop production of tea and other products, okay, hill areas. Webel, we have recently had a very good discussion with the MD of Webel. I had a very good discussion with the Principal Secretary, Information Technology, Government of West Bengal, and okay, that means, and also our Vice Chancellor is connected with all these activities. So this is a good activity. We're looking forward, and obviously we're looking for non-profit organization. We're trying to have some collaboration with Feeding India, a non-profit organization who can help us in actually gathering the data, understanding the problem, right? And I invite all of you who wish to contribute in this technology aided. I will say that technology is the aid, but the problem is in the domain of nutrition and food security and food safety. So whoever wants to connect and collaborate, right, I will be very happy. And again I say that I have just tried to pitch you the pitch you some of the things in technology, technology challenges and technology directions, right? And there are a lot of open problems out there in terms of research, right? And I hope that I have created some food for thoughts for all of you. Thank you. So I end up here.
Yes, sir.
Thank you,
Thank
sir,
you, sir.
for this wonderful deliberation, bringing in all the things and giving us a glimpse of how technology can actually influence the food security, which generally is taken or seen from a nutrition point of view, or sometimes from the economic point of view. But in the times of changing technology, this viewing of the problem actually should be changed, and you have given us very nice food for thought. Bipsha, you can continue.
Yes. Thank you, sir, for sharing such an informative and insightful lecture with us. Is there any question from the audience?
I have a question. You have told many aspects. Actually, about 3 to 4 years ago, I gave a project for that is the banking of food bank, and that is different specific area based foods, that is in the local area. There are many foods that are taken by the local people. That is I can name one, that is googly. It is taken by tribals and villagers throughout 8 months of the year, and they get mostly the protein from that googly. So these are not in the ICMR data book. These type of datas, they are not included at all. There are many such food, and during this pandemic I contacted Professor Parthopotim Babu, who is a very good genetics ISER genetics and genetic. He told me that I was thinking that with the people of China, they have this disease COVID, but the eastern region of India, they are of the same genetic population, and the genetics is very similar to the Chinese people. But he told me that, and he said, so then I asked whether there is some food in the northeastern part that may have protected them a little better than the from Chinese people. And he said that yes, this type of there are many such food items in eastern part of India, and that can be one of the reasons where it may have protected from COVID. So we can also do this type of project where big data can be used for and to make the bank of different food items, and which are very locally available food items, not very common throughout India.
Yeah, this is a very good input, Dr. Professor Pubali. Yeah, this is a very good input, and actually that's why we require the domain experts to be in this activity, because because this particular side of the food, that is the food itself, can actually can say influence the health, right? And see, that is our technology is again I'm telling, our technology is an aid, right? We are trying to look into that, obviously, as you said, that how we can analyze the data and how we can converge to a sort of sort of predictive analytics or prescriptive analytics, that okay, based on the data, based on the domain knowledge, okay, we are trying to just generate a type of simulation engine or a model, okay, which will give these insights. And I think these are the real thing which we need to have to okay, that means to get into this particular
Because
activity.
I'm
Yeah.
working with human samples for a long time, and we find that ICMR, there is very little data on Indian population. We always use the hemoglobin content of the world average, but the same thing when it comes to India, it shows that always the whole female group, they are suffering from anemia. It may not be this that we may not be suffering from anemia. It may be that is our normal average of hemoglobin, because when NIN, National Institute of Nutrition, they are working to increase the hemoglobin content for about 20 years, giving iron tablets to specific villages, but not a single unit has increased. The hemoglobin content has increased. Maybe that is our normal data, normal hemoglobin content. So we don't have these parameters on the basis of Indian population. These parameters, always we use the world average population. So I think and like mother's milk, the compositions of mother's milk, the lipid compositions, they are all specific to a specific country, to a specific dietary habit, that influence those types of data, and we don't have these in the data book of ICMR or specific for India. So we can work with many things, I think.
Sure. We should start our
Yes.
interdisciplinary research, and maybe the students. I have formed a group. We can have a group meeting with your group, or with your research, with the research group of the student group
Yes.
of your department.
Yes.
And I think this will be a very good activity, and we will surely at least try to solve at least one problem. Okay.
Yes, I have many
But-
problems.
Sure.
Any more question?
Sir, in a country like India, there is another big challenge that wherein you people are doing a great job in using the technology in the positive sense. There is again this various divisions in the society, specifically economic divisions, which actually causes exclusion from of the majority of the people from the benefits of the technological advances. This is again a very big challenge whenever we are considering India as a nation, I suppose.
Yeah, very well said, Professor Purba. Actually, this is also a challenge, and we are trying to see that what are the so the way we now think about technology is that we first try to see that what are the reach out of that population, okay, in terms of technology. Right. Obviously, the technology reach out of the population is increasing. It's not stagnant, though the rate of increase, okay, is not the same. The good point is that means the communication infrastructure, which is already started by it's called rural digital connect, okay, from Government of India, and very soon, within 2 to 3 years, the broadband, the optical broadband will connect, okay, up to the gram panchayat. Okay, so all the gram panchayats in India, it's called the R-Bharat, Rural Bharat, Digital India Mission. Okay, and that will connect the gram panchayat. Now the challenge is the last mile, which is the most challenge, right, from the panchayat to door, okay, to the rural areas, right? And there are some technologies, some low-cost technologies which are available. CDAC is working with, okay, in tandem. We are also, that means, as you know, that I also have some activities in multiple groups apart from universities. I'm also a part of that Ministry of IT and CDAC activity in terms of the connect, that is a last mile connect to the villagers, in terms of it's a new technology. I think some of you know that it's called a 5G 5G wireless technology. So once this type of technologies can cover the rural population of India, right, I think more digital services can be offered, right? And then our aim is to how to take the digital platform and how to support the okay, this particular aspect of okay, of human life, which is the nutritional support and food safety and security. We don't know that where we will lead, but we are trying to okay, go in small steps, such that, okay, assuming that the digital reach will be better and better of the rural population also within a span of 3 to 5 years. Yeah.
Wonderful, sir. Thank you so much. It was such an
Very
enlightening
much
lecture. Specifically, the advances in technology and what technology can do. You had given a very beautiful glimpse into the future, which was very encouraging for all of us, for the fraternity as well as the students present here. Thank you for motivating us, and please keep some of your precious time in future also to share
Sure.
your experiences of the field with all of us. Thank you so much.
Sure.
Yes.
Sure, Professor Purva. Sure.
Okay, okay. Thank you.
Thank you, sir, for sharing your valuable time with us. Thank
Thank
you.
you. So I am leaving. I have a meeting starting at 7. Okay.
Yes,
So
yes,
so
you are a
so
very
thank you all. Thank you.
okay.
Okay.
Now we will move on to our next session. It is poster presentation session. Poster presenters, please respond.
They have responded, Bipsha.
Okay, I will
Just
start with Onushwa.
yes.
Okay. I would request Onushwa Singh from Department of Physiology, University of Calcutta, to present her poster entitled Kulekhara Herbal Tea Approach to Cure Anemia. Onushwa?
Good evening, everyone. I am Anushwa Singh from Department of Physiology. I am working under Dr. Raghavendra Mishra and Dr. Roshanara Mishra. Am I audible clearly?
Yes, you can share your screen and
Okay.
proceed.
Yeah. Is my screen visible?
Yes.
Okay. So I am starting my presentation. Good evening, everyone. Today I am going to present my poster entitled Kulekhara Herbal Tea Approach to Cure Anemia. Anemia is a serious health issue, but though it is neglected worldwide also, iron deficiency anemia is the most prevalent, followed by anemia of inflammation. It affects almost the children and women of all the ages and also is evident in men. Anemia can be caused due to low dietary iron intake, impaired iron absorption, increased iron demand. In case of pregnancy and menstruation, anemia is also evident. In case of inflammatory condition, iron absorption is also seen. The conventional treatment of anemia is iron supplementation, but there are several complications associated with it. These complications may include the gastrointestinal related side effects, poor gastrointestinal absorption, poor intolerance, and the poor acceptability among the people. So this gives us the fertile ground to test the potential of herbal remedies in curing anemia, as herbs are used since ancient time to cure various ailments. Depending upon the ethnobelief, one such medicinal plant is kulekhara, which is used to cure various diseases and anemia as well. The traditional method to use kulekhara is to boil the leaf and intake the strained water. But it is a tiresome process and kulekhara is not found everywhere. On the other hand, herbal tea is getting more popular nowadays. So this gives us the idea to develop kulekhara herbal tea and to increase its popularity among the people. So to achieve this, our aim is to process the kulekhara leaf and develop the herbal tea from it, and finally check its bioactivity among the different anemia model and human volunteer. Here in this study, we have used iron deficient anemia model. Firstly, here we have prepared kulekhara herbal tea using various processing methods and checked its different comparative analysis of different components and done its sensory evaluation. Here we have found that the 6-hour withered microwave dried kulekhara herbal tea extract gives the best result, and we have done the HPLC analysis of that processed kulekhara herbal tea. Next, we have checked its bioactivity in the iron deficiency anemia model, which was developed by iron deficient diet induced in mice model. It was found that all the hematological parameters such as hemoglobin concentration, total RBC count were altered after the treatment. It was also found that serum iron concentration and TIBC parameters were also normalized after this treatment. Next, the kulekhara herbal tea was given to the human volunteers, where we also get a positive result. Hemoglobin, RBC count get increased after kulekhara intake. Serum iron was also increased in the volunteers. So, from our study, it can be concluded that the bioactivity of kulekhara was preserved in herbal tea formulation and effectively prevent or cure the iron deficiency anemia in human volunteers and experimental murine model. A detailed study in a larger population is warranted to understand the efficacy of kulekhara herbal tea and the mechanistic basis of its hematinic effect. Thank you.
Thank you, Anushka Singh, for such an wonderful presentation.
Thank you.
Now I would like to request Aishwarya Aich, Junior Research Fellow, Department of Physiology, University of Calcutta, to present her poster entitled Kulekhara, a Key to Combat Anemia of Inflammation.
Okay.
Share your screen and you can start.
Yes, I am sharing. Okay, is it visible?
Yes.
Okay. So, good evening, everyone. I am Aishwarya Aich, and currently I am pursuing PhD from Department of Physiology, University of Calcutta. The topic of my today's poster is Kulekhara, a key to combat anemia of inflammation. Actually, the last presenter is my love mate, and she has already described that how the anemia works and all these things. My work is the continuation of the previous presentation. She has already told us about the anemia, the problem regarding its treatment, also about the Kulekhara tea bag developed in our laboratory as an alternative strategy. So in this study, we wanted to investigate the particular mechanism through which Kulekhara can prevent anemia. Iron homeostasis actually plays a very important role in the formation of anemia. Anemia of inflammation is mainly seen in people who have inflammatory conditions. Now, it is very difficult to basically differentiate the anemia of inflammation and iron deficiency anemia. And these two conditions even can coexist also. So, when any inflammatory condition occurs, the immune cells releases pro-inflammatory cytokines, especially interleukin 6. In normal state, this iron, our body iron can comes out from cells to bloodstream through ferroportin, which is the solely known iron exporter. But in inflamed condition, IL-6 stimulate hepcidin production. Now, these hepcidin block ferroportin, causing lower iron level in the blood but high iron load into the cell, and thus it causes anemia. And it also disturbs the iron homeostasis as well. So, this pathway is very important undoubtedly for anemia formation. Our study aimed to find out the pathway through which this is the aim of our study. We wanted to investigate that through which Kulekhara aqueous extract inhibit anemia. And we targeted IL-6. It is the key upstream immunomodulator of anemia of inflammation. It was chosen as our protein of interest, and we wanted to know whether the components of Kulekhara leaves can interact with IL-6 or not. Then methodology. So why in silico study? Why not in vivo study? The thing behind is that in vivo, in vitro studies are more expensive, more time consuming, as well as it harms animals. We need to sacrifice them. But in silico study, it is more convenient. Okay. And the most important thing that it can give us an idea about the probable result. So after that, getting that result, it can direct us that in which path we should go, and we will do the in vitro in vivo study later after getting the idea from it. So for this study, we used Schrodinger software. We took human IL-6 protein structure. It was 1 ALU and it was reported in RCSB PDB. We prepared it in the Maestro, that is in the part of Schrodinger software, and Ramachandran plot analysis was done to validate the structure, and it was fine good. After that, CD analysis was also done to find out the important residues, and the ligands were also prepared. Ligands were nothing but the Kulekhara components which we got from the HPLC analysis, as my previous presenter described. So it was done by her, and these components were docked with the IL-6, and it gave actually a very good result. You can see that the more negative the docking score is, the more good it is. So it was found that rutin followed by naringin and myricetin give a very good result. 12 out of 20 components give a very good affinity, and they interacted with glutamate 55, alanine 56, and glutamate 59. These were also indicated important residue as per CD analysis. So it was also finding good for us, and from all this result we can conclude that yes, there is a high possibility that Kulekhara compounds are able to inhibit IL-6, and maybe through this pathway, by inhibiting IL-6, it suppresses the downstream pathway leading to anemia. But as I said, in silico study have some limitations also. So before concluding, we need to do the in vivo study and in vitro study as well. Even as a future plan, we have planned to check the in silico study of Kulekhara components with hepcidin and IL-6 receptor also, to get the more clear view about the mechanism. Thank you.
Thank you for such an informative presentation.
Okay.
Now I would request Vasundhara Chand, Research Scholar, Lady Arwin College, Department of Food and Nutrition, University of Delhi, to present her poster entitled An Assessment of the Benefits of Exclusive Breastfeeding with a Focus on the Controversies to Feeding. Vasundhara? Yes, you can present.
Good evening, respected judges and my dear co-participant. I am Vasundhara Chand, research scholar from Lady Avan College, Delhi University, New Delhi. Today I am going to discuss about the assessment of the benefit of exclusive breastfeeding with a focus on controversies to the feeding. Breast milk is the best source of nutrient for an infant, and it is universally endorsed by the World Health and Scientific Organization as the best way of feeding infants. Many studies have highlighted the innumerable benefits of breastfeeding, and as per the World Health Organization, infants should be exclusively breastfed for the first 6 months, followed by the breastfeeding along with the complementary foods for up to 2 years of age or beyond. The breastfeeding helps in lowering the risk of gastrointestinal infection, pneumonia, and urinary tract infection among infants, and it helps in reducing the risk of type 2 diabetes among mothers. It has been noted that the mother faces the infant feeding problems, and with this, the objective of today's presentation is to examine the advantages of feeding exclusively and to give an outline of the issues during the breastfeeding. So when we talk about the express breast milk, so when the mothers are unable to breastfeed their infant, so they wish to express their milk using the hand or the manual or electric pump. The reason why the mother expresses the milk is when the baby is immature, unable to suck, resumptions of work, nipple are too sour to feed and to increase the supply, and for the comfort during the sudden weaning and separation or engorgement. The pitfalls of express breastfeedings are that there is no skin to skin contact. It's better with less handling. There is an absence of when there is absence of baby efficient suckling, and it requires more work, so more cleaning is required in it. So when you talk about the composition of the breast milk, human milk is species specific and is superior to any other breast milk substitute. It contains the factors such as immunoglobulin, T lymphocyte, enzymes such as lysozyme and phagocytes, and there are 3 phases of milk, namely the colostrum, transitional, and mature milk, with each with distinct characteristics. So, coming to the contraindications to the breastfeeding, in infants who are special health problems such as galactosemia, maple syrup urine disease, phenylketonuria, and among mothers who have untreated tuberculosis disease or are human T cell lymphotrophic virus type 1 and type 2 positive, and mother who are using the drug of abuse and are receiving the or some diagnostic or therapeutic radioactive isotopes or chemotherapeutic agents, and mother who are HIV infected. So with this, we can conclude that the cost-effective way of reducing the risk of disease such as obesity, hypertension, eczema, type 2 diabetes, as well as mortality, breastfeeding is very important, and there is a need for nutrigenomics and nutrigenetics based research in this area. So thank you all.
Thank you for such a wonderful presentation. Now I would request Taposhi Pole, Senior Research Scholar, Food Technology and Biochemical Engineering Department at Jadavpur University, to present her poster entitled Statistical Optimization of Alkaline Protease Using Isolated Strain. Taposhi?
Yes, I am audible.
Yes, you can share.
Okay. Just 1 minute. The screen is visible?
Yes.
Yes.
Just rotate your mobile.
Okay.
Yes.
Good evening, everyone. Good evening, respected ma'am and everyone. Myself, Taposhi Pole. I am the Senior Research Scholar of Jadavpur University, Food Technology and Biochemical Engineering Department. I am completed my PhD in Jadavpur University. Today, my topic is statistical optimization of alkaline protease using isolated strain alternator USFQ1. First is introduction. Protease is the alkaline protease is the hydrolytic enzyme which catalyzes the cleavage of peptide bonds. This enzyme used in 60% in industrial uses, and this is the very much industrial uses in recovery, pharmaceutical, detergent production, food industry, and my optimization was carried out by statistical process through the RSA. RSA means response surface methodology. It is a quick process for optimization of the large number of the variable, and this is my materials and methods where shows the microorganism is isolated in alternate USF1, and the fermentation was carried out is the optimization of the fermentation done with respected the incubation time, temperature, age of inoculum, volume of inoculum, and agitation of the media. An enzyme assay was described by the Kempf method, and the protein assay by the Lowry-Attas method. And this is the regression equation where y is the responsible variable and beta 0 is the intercept, and beta 1, 2, 3, 4 is the interaction coefficient, and A, B, C, D all are interaction, and this is the most important in our studies. And this is the result and discussion where shows the statistical optimization approach using the RSN was the most important factor, and by the employing RSN, the maximum protease production was 6150 mg per gram was achieved in 7 days incubation, 30 degrees centigrade, 30 ml of volume, and 7 days age of inoculum, and 120 RPM. And this is the 3D model which shows the interaction everything, and the green zone is the maximum production and the blue zone is the lower level of the production, and this is the interaction. And the conclusion, the specific activity of the found the 200 unit per mg was optimized one factor time in the calculated 601 unit per mg by optimized by the statistical approach, and this is the most important in our industry, and this is a valuable, and this is our reference, and thanking you.
Thank you, Taposhi, for such an informative presentation.
Okay.
So with this presentation, our day comes to an end.
Yes.
Yes, Viksha, so you conclude.
With this, we come to an end of our session for the 4th day. I would like to remind that we already shared the link of feedback form in the YouTube chat box and also the Telegram group. The feedback form will be open till 8:30 p.m. and the certificates will be distributed to the participants accordingly. Now I would like to hand over the microphone to Dr. Annalakshmi Chatterjee ma'am to conclude today's session.
Thank you, Bipsha. Thank you everyone for attending today's webinar lectures. We hope you enjoyed your experience and learned valuable informations regarding in the field of Helicobacter pylori, as well as new insights into the technology involvement in the food safety and quality improvement of the food, which you can implement in your future research works. I would like to thank our speakers for today also, and the audiences. Thank you very much for joining us. We are looking forward for your cooperation for tomorrow's session. Thank you everyone. Okay, tomorrow at what time we will start? Just give an information.
3 o'clock.
Okay.
So say 10 minutes before that we will be joining.
Okay. Thank you. Thank you. I think we can end here, isn't it?
Yes, we can end here.
Yes, we can.
Thank you everyone.